Okay, well, thank you very much, Eric and Matt, for the introduction. So just a few housekeeping items. Well, we're going to spend 4 hours today, 4 hours tomorrow. I hope you can stay for the whole thing. We'll have a break with cookies and coffee. So for those who are here in person, we can get that break. Basically, we start with a little bit of introduction in terms of general history of genome sequencing and assembly, a little bit of applications, key concepts that are important, and then we dive into the sequencing technologies. And then we'll get started with the practical part. So we'll get you set up if you haven't already created your own account in Galaxy. We'll make sure you have your own account, and we'll get started with the practical. And we will have presentations intertwined with the practical, where we explain things like K-mers, how you evaluate genome assemblies, and so forth. So, okay, how do I?
Look at the second desktop.
I have to move my pointer here. Okay.
Yeah, because this is.
Oh wait, can I just see it here as well, or
What?
can I see it here so that I don't have to turn to the left?
So do you want to just mirror this completely?
Yeah, I think it's much better.
But then you want to have this separate.
But I don't. Oh, the
You asked for this separate.
No, it's just
Okay.
for the beginning.
Oh, okay,
yeah.
sure.
Sorry.
Technical difficulties.
Yeah, otherwise.
Or you can just bring it.
Well, I'll just start with a few demographics of the participants. They don't see what? Oh, they don't. Yeah, because now it's
I'm
not
not
shared.
done yet.
All right. Yeah. Just a second.
And there are a bunch of notes in the chat as well.
Are there notes in the chat, like something we should? Something relevant? Okay.
Let me, wait, I didn't share the screen again. Share screen, current desktop. All right. I forget how to close this.
Now a slideshow,
Yeah, I'm just trying to get this to close.
but I think it will disappear. No,
No.
will not. Seriously?
Click it again. Hide.
Oh. Okay, hopefully you can see it online. You can see it here. Just really quickly, demographics, most of the participants are postdocs, some research assistants, graduate students, staff scientists, other, I don't know if you want to say what that means, but yeah. We have people with some coding experience, but mostly with no prior coding experience, apparently. So never done programming before. We have people that are familiar with like Illumina mostly, short read sequencing, but some also with PacBio sequencing. Interestingly, some people with BioNano. Hi-C is very important today in genome assembly, so we'll cover that. ONT, that's also an interesting technology for many applications in genome assembly. And then we are going to use Galaxy as a platform to essentially assemble our own genome. And it's interesting that several people have heard of it, but not used it, and even some actually used it. Maybe we'll have a chance to tell us how you've used it so far. Well, the question here is whether you have used reference genomes for your own research. It seems that many have. Sometimes human, sometimes model organisms, not human, and some other times non-model organisms. And again, it will be interesting to know, especially I would say for non-model organisms, what kind of species are we talking about? Finally, how and why have you used the reference genomes? Mostly comparative genomics and functional genomics, some GWAS, prop design, genome editing, PubGen, and so forth. Just one thing, we have a Slack channel that was set up when we first taught this course a few years ago. So I don't know, this is probably not the best way to share it, but maybe if you can send it by email, Matt or someone. And we can use this, to coordinate today, tomorrow, but also for any questions you may have in the future. Okay, so I'll dive into the sort of a little bit of a history of, let's say, DNA sequencing and genome assembly that are really two things that have been intertwined in the history of of bioinformatics. So, well, first of all, I mean, I don't have to really explain why this is important, but even in the 80s, Frederick Sanger, the inventor of the Sanger sequencing, acknowledged that this basically, it's really important to get the sequence and to study the sequence to understand living matters. And of course, we have plenty of applications. I mean, we have already mentioned many of them in the survey. Population genomics, GWAS, comparative genomics, conservation genomics, epigenetics. You name it. So what we're after is this reference genomes, that we can describe briefly as contiguous. So we'll see what that exactly means. Complete. Again, we'll see what that exactly means. Accurate sequences that are representative of a species, and then it means that we can look at all the genomic elements, single nucleotide polymorphisms, copy number variants, mobile elements, segmental duplications, centromeres, you name it, depending on the scientific question. So there are basically three recognized sort of steps in the evolution of sequencing techniques, and again, where the genome assembly perspective tends to used to evolve in parallel with those. So you have first generation sequencing, second generation NGS, and third generation. So of course, everything starts after the discovery of the structure of DNA in 1953. What actually interesting is the same year, Frederick Sanger, so the guy over there, was also able to describe the first sequence of a biological molecule. And that was not DNA, that was a protein, because it was easier at the time to sequence proteins. And actually he introduced the same principle that we now use for assembling a genome or looking at DNA. So you chop the molecule into pieces, then look for, imagine this is the original molecule I have here, in this case of a protein, and you have all these fragments. Now, after of course, you have multiple copies of the same molecule. You chop it into pieces, you have multiple fragments, look for these overlaps between them, and then reconstruct the original sequence. I'll just go very briefly over this, but like in 1965, we have the first nucleic acid sequence that was actually assessed. Now that's alanine tRNA. We're talking about 76 nucleotides, required 5 people working 3 years on 1 gram of pure material isolated from 140 kilograms of yeast. So not very efficient at the time, but of course we got better. 1968 is the first DNA molecule sequence, because that one was nucleic acid, but not DNA. So in this case, it's the cos site of the lambda phage. I won't go into details here, but the lambda phage has this sort of circular and linear nature, depending on the life stage. And essentially, the cos site, can be opened by these terminates, and then leaves overhangs. And because you have overhangs, now you can use the overhangs and you can sequence the overhangs, which was done at the time. So we're talking about 12 base pairs. You can try add, with a polymerase, you can try adding 1 nucleotide at a time, see if it fits. If it fits, then it means you understood what base was at that particular position, and then you try adding a new nucleotide and so forth. Of course, laborious, but again, gives us a sort of first approach that was then turned into what's called first generation sequencing. We have a few more stuff that gets sequenced in the 60s and 70s. So we are increasing the length, but we're still very short. And then we get to when Sanger really start to work on DNA sequencing. So 1975, develops this plus and minus method, and uses it to sequence the first entire bacteriophage, FIX 174. Which is 5,000 base pair long. It's actually funny because this is the control in all Illumina sequencing experiments today. When you run a Illumina, you will always find this in your results if you try to dig in, because it's the positive control that actually the sequencing reaction works. I won't explain how this works, but then we have another few methods like 77, the Maxim Gilbert method, which is purely chemical. It's still the only pure chemical method to sequence short nucleotides sequences. And then we, of course, get to the Sanger sequencing, same year. I'm sure you have already used or learned about this many times, so I won't explain how this works, but essentially you have this deoxynucleotides, you have a mixed reaction, and then you look on the gel or in a capillary electrophoresis out the differential migration tell us about the pattern and the original sequence. 79 is also a very important year, maybe the birth of bioinformatics in a way. It's when Roger Staten invents, let's say, the first DNA sequencing software. And that was a powerful approach because in 82, Sanger uses it to assemble the 50,000 base pair long bacteriophage lambda entirely. So again, this is the real shotgun sequencing approach. So basically, you have to take your own DNA molecule, you fragment it, you sequence it, and you do this in parallel. So that was the sort of breakthrough. All the stuff that was done before was, laborious, and every step here, you can clone all the sequences, sequence them in parallel, and then again, find the overlaps and reconstruct the original sequence. Of course, then we have, PCR that comes out in 1983 that, of course, allows you to access the DNA more easily. 80s and 90s, we have automation of the Sanger sequencing method now with laser beams. Oops. I got it. Laser beams, detectors. Well, in parallel, we have computers that are developed and so forth. And now we get this kind of chromatograms that we can use even today. Other stuff that it's developed around the same time, now 1988, the pyrosequencing using the luciferin. It's a different kind of reaction that also leads to sequencing essentially. And, it's a blossom of data in the public database. Now, this is the curve describing the increase in log scale of data in GeneBank. And of course, around the year 2000, you start to also have whole genome sequencing after the completion of the Human Genome Project. A few other things in 1990s. Now we have paired-end sequencing, which is now like common and the standard for Illumina sequencing, where you read all the fragments on both ends. This is desirable for many applications, including for reference genome assembly, because then you can span repetitive elements, as we will see. And you have a lot of course, bioinformatics improvement. We have development of BLAST. We have the development of the so-called quality score, the Phred score, which is this general attribute that is often used in many instances. Is used to measure base calling accuracy in sequencing reads. Is used to measure consensus accuracy in genome assemblies and so forth. Now it's a log scale, so if you have a Q score, Phred score of 10, it means that there is one error every 10 bases. So 90% base call accuracy. If you have a Phred score of 20, then there is one error every 100 base pairs, 99% base calling accuracy, and so forth. So it goes up by one order of magnitude every time. Okay, so what is really leading us into what's called next generation sequencing is the 90s with the Human Genome Project. So at the end of the 90s, you have in 2001 particularly the first draft of the human genome with Bill Clinton having Craig Venter on the left and Francis Collins on the right, announcing it at the White House. Actually interesting that the human genome was never really completed, and only this year, combined effort, which we also participated into, led to the completion of the human genome, particularly for the complex parts that are more repetitive and were not resolved until recently with some of the technologies that we're going to use that really made a difference. Here, I will just highlight the fact that there were two competing efforts. So one was led by Craig Venter, the guy on the left, and the other one was led by the NIH, led by Francis Collins at the time. And they were differing in the approach. So one is called hierarchical shotgun sequencing, and that was the public one. And then the other one is whole genome shotgun sequencing. And that was the Celera approach by Venter and his group. So the difference is that in the hierarchical case, they were using bacterial artificial chromosomes or yeast artificial chromosomes to integrate big chunks of DNA into it turns out that this bacteria can actually harbor very big chunks of DNA. And then they were sequencing all these chunks separately and then trying to find the overlaps between them to reconstruct the final sequence. This leads to a easier to solve problem because the chunks individually, they are smaller, but of course it's more laborious because you have to clone all of them, reconstruct all of them, find the overlap. So especially that's why it took them 10 years essentially to get there eventually. Whereas the approach that Venter was following was this whole genome sequencing. Now, so basically you don't have any intermediates in terms of backloads. You just sequence everything. And then try to reconstruct bioinformatically the original sequence. It turns out that this was the most successful approach in terms of the quality of the final sequence, but the approach that it's now what we use today, so nobody's using BACs anymore or artificial chromosomes anymore, but the ones we're using today is whole genome sequencing. And at the time, nobody really believed that was possible for large genomes, so they were proved wrong. So, yeah, of course, then the completion of the human genome means that essentially, everybody's investing now into the sequencing. So you have a drop in cost and an increase in throughput. There is this institute, the NHGRI, that is funded specifically to support the new sequencing technologies, the development and so forth. Yeah, this is a very, very significant increase as represented here. So that's how we get to second generation sequencing. So even the human genome was not completing using anything that's close to Illumina. It was essentially Sanger sequencing at scale. Then we have what's called next generation sequencing. So I'm not going to go in details now, but because we're going to discuss this with Olivier, but the idea is now that you can parallelize the sequencing a lot using these flow cells at the nanoscale and basically generate millions of reads in one go. This was initially commercialized by Solexa and then purchased by Illumina in 2007, which is now the leading company in the world for sequencing, at least with short reads. This allows to generate a lot of new genomes, even for non-model species. This is a special issue that was out in 2014 by Eric Jarvis and others, comparing 50 or more or less 50 avian genomes. And again, these are all non-model species, and it was possible to do so because of the the reduction in cost and the increase in quality of the short read sequencing. So this is, for instance, now you could start to reconstruct the avian tree from a whole genome perspective at that point. Then we get into third generation sequencing. So the key difference is that we go from this surfaces where you have a process that will go a little bit more into later of bridge amplification, where essentially you amplify the signal so that you can visualize the bases and infer the sequence. We go through third generation sequencing to single molecule. Essentially, all third generation sequencing technologies are single molecule technologies. These leads one way or another to longer reads. We're talking about 100 to 300 base pairs for NGS, whereas in all third generation sequencing, we're talking about at least 10 KB long reads. The throughput used to be lower for third generation, but it's getting higher and higher, as we'll see. And also the quality used to be lower, but it's getting higher and higher. So yeah, in 2014, Illumina had 70% of the market, 90% of data produced around the world. I don't know if this is still accurate. In 2019, but probably it is, if it's not even bigger. In 2019, Illumina tried to purchase Pacific Biosciences, which is this other technology that we will hear about. And I would say this is when third generation sequencing really proves to be, the future of DNA sequencing. A few things about, just sort of housekeeping stuff that you should be aware of. One is, aside from the historical perspective, few key concepts. So one is that sequencing coverage, depth. I'm sure you probably all know about this, but just to get things straight. So it's the average number of reads that cover a known reference space. That's one possible definition of it. This is very important because when you do whole genome sequencing, you are sequencing randomly the molecules in your pool of molecules. So you will never be able to sequence each molecule exactly once, as you would do, for instance, with Sanger sequencing. So you need essentially to sequence a lot more so that on average, that's what you get for each base. And if you sequence, let's say 30x, then of course it means that some bases will be sequenced less than 30x and some other bases will be sequenced more than 30x. And particularly there are biases in sequencing coverage, which essentially means that because of the intrinsic nature of some regions of the genome, some of them will be sequenced more or will be sequenced less than what you would expect. And of course, there are also potential biases that are introduced in terms of library prep. Now, when you generate the library preparation, when you generate the libraries for sequencing, that will also affect this. Introduce bias. It's very well known for the GC bias for Illumina sequencing. Here is the Poisson distribution that we usually expect when we sequence a sample. This is 15x coverage. And of course, it means if we sequence a sample at 16x, 15x, there are some regions that are, let's say, 10x, some other are 20, but there are some that are 1x. And we can expect that there are some that are 0x coverage because again, it is a Poisson distribution, is a random process. So you get not to, just by chance, you don't see some of the regions. So the idea is that the more coverage you have, the more this distribution shifts towards the right, and the less likely you will have regions of the genome that are covered 0 times or too few to be accurate. Other key concept, repeats. Genomes are full of repeats. These were the last bits of the human genome that were not resolved since 2001. And the repeats are a problem because they break your contiguity of the genome. So imagine this is the original sequence, now the black line, and the red here is a repeat copy, means that essentially the same sequence repeated exactly twice in the genome. So you have one here and one there. These are the sequencing reads. So you can see that the sequencing reads essentially do not cover, do not span the repeats. None of them is really spanning from one end to the other end, the repeat copy. So you don't know how to really reconstruct the original sequence. You end up having to break down your assembly into contigs, not a single sequence, therefore. So 1 contig, 2 contigs, 3 contigs, and 4 contigs. And this is a collapse repeat consensus contig. So it means that essentially it took reads from here and collapsed them into a single sequence. So that's why long reads are actually important. Because essentially they improve your chances of reconstructing a contiguous assembly. Also, they are important because if you're actually looking at not a single reference genomes, but many individuals from the same species, for instance, they increase your chances to, for instance, detect deletions, insertions, duplications, inversions, translocation, expansions, all this kind of structural variants that with very short reads are essentially impossible to resolve. And just to give you some idea, we have, if you take any 2 humans, you have more or less 5 megabases that are, distinguishing me from Olivier, for instance, that are single nucleotide variants, so point mutations. But then you have 3 megabases of indels, short, repeats, very short repeats like triplets or homopolymers that are also distinguishing me from Olivier. But then if you look at structural variants in terms of the number of bases that are affected, we're talking about 10 megabases. So even if the sequence is the same, you may have an inversion that essentially leads to millions of bases being inverted with respect to, for instance, me and Olivier. And if you take another example, another comparison, human versus chimp, we're talking about 70 megabases overall. And again, we see the same pattern now where essentially there are more bases affected by SVs than the rest. I won't describe this, but This is one of my preferred examples of how structural variants are actually really important in terms of determining phenotypes. So this is the ruff, which is a bird you can find, for instance, in the Scandinavian region. And essentially, you have three male morphs: the independent, the satellite, and the feather. And they look very different. No, they're all males, but they're really, really different. And it turns out that there is one single structural variant that has been, out there for several million years that essentially is causing these different phenotypes. And it's an evolutionary stable variant, meaning that is essentially, it can be sometimes. If it's, for instance, if it's homozygous, it will not be viable, but it's essentially stable. So it means that because of the strategies that the three different male morphs have evolved in terms of reproduction, it can survive over the years. And of course, I mean, being able to detect this, requires a good reference genomes on one side and sequencing multiple individuals with different morphs on the other side. There will be a little short video demonstrating this, but maybe we can keep it for later. So the last thing I want to mention in this introduction is sort of who we are. Maybe I should have started with this, I don't know, but who we are. So the VGL, so the Vertebrate Genome Lab, is a group. We are in this building here, but we're part of a larger project, the Vertebrate Genomes Project, that has three main sequencing hubs. So one is here at Rockefeller, the other one is the Wellcome Sanger Institute in the UK, and the Max Planck Institute in Dresden, and many other collaborators around the world. And the aim is to generate near error-free reference genomes for all 70,000 or so extant vertebrate species. We're working this in phases. So phase one, that's where we're at now. Hopefully we'll be able to complete it next year. It's representing each order of vertebrates with one reference genome. We're talking about 200 or 300, 260 or more, a bit more species. Then we'll move to families, genera, and all the species, hopefully. And we deposit everything we generate in this repository called the GenomeArk. So it's a publicly available public database, open access, a library of genomes. You can see an example here. So if you just Google GenomeArk, that's what you will see. So you have the species, and we deposit everything. We deposit the raw data, the intermediate files, the final assemblies, almost as soon as they are generated. And so they're accessible by anyone. And this is also how we exchange data between the different sites in the consortium. And I think this is a powerful concept now, because really we are making an effort towards open science and open data. Just briefly mention our goals is to, again, of course, use these genomes for understanding biology. So we want to reconstruct the phylogeny of all vertebrates, study chromosomal evolution, comparative genomics, disease-resistant genes, and of course, help conservation through conservation genomics. Again, we're hopefully going to be able to complete this next year. This is a little bit outdated, but yeah, we're getting there. And I'm just gonna finally mention that this is one initiative, was probably one of the very first initiatives, especially using long reads of its kind. And now this is becoming more and more widespread around the world. So these are a few of the initiatives that are ongoing around the world that target reference genomes for biodiversity. Some are focused on specific regions like the Darwin Tree of Life for the British Island, or more generally for Europe, the European Reference Genome Atlas. Some others like the VGP are focused on some specific taxa. And overall, they all go under this Earth Biogenome Project umbrella, which is a coordination network for all this project that is really basically trying to complete all 1.8 million species of eukaryotes in the next 10 years. So with that, I will leave. First of all, I don't know if you want to have some questions or if this is all clear. also from the chat on Zoom. If not, yeah, Eric.
Well, that question just say, I think it's good to clarify another reason for why you need coverage, why you need to sequence the genome 30 times over.
Yeah.
So I was trying to get those that one sequence as well.
Should I clarify it? You have it in mind. So what is the other?
Well, it's to not every sequence read you generate, even though it's 99.9 whatever it's accurate. Once you have 3 billion base pairs, there's a lot of inaccuracy.
Oh yeah.
So you need to sequence multiple readings and take the consensus of those readings together. And the higher coverage you have, the higher accuracy you get. Right.
So for those online, I don't know if you heard this, but basically the reason to sequence more is also because the every read that you sequence will have some sequencing errors, again because of this because everything is not there is nothing perfect in the world, and so even sequencing reads do contain some errors, and if you only have one read from one region, then you will have no way to disentangle the errors from the actual correct basis. But if you have multiple reads that span the same region, then you can use them in their sort of consensus to reconstruct the correct base in the genome.
And the second thing I was saying, the indel was that because not everybody would know, but in insertion deletion.
Yes, indel, insertion, del, deletion. So short polymorphism that have a variation in terms of the number of copies, essentially, but like triplets or dinucleotides, stuff like that. Okay, yeah, I'm sorry, I cannot see if there are any questions or help.
Just answer the thing real quick.
Oh yeah, here is the chat. Okay, perfect. Okay, cool. So with that, I will leave it to Olivier for an overview of the sequencing technologies, and then.
So I promise you there's a last part before we do really some bioinformatics. Hope so. We thought it would be very, very important to understand what is behind the data. Because as Giro was saying, some data have biases, some others have different type of biases. And having a good understanding about how this data was generated is important to address problems you will have doing genome assembly. But before starting, I wanted to show something. So Giulio talked about the Human Genome Project sequence with Sanger, and this is how Sanger sequencing looks like. So this is a 96 well plate. Sorry for people on Zoom, they can see it. I don't have a camera on, so.
I'll turn it on. I'll turn it on.
It's just for Jim. Sharing this thing. Share video.
All right. All right. You can see. Yeah, I'm big there too. Okay. Thank you. All right. So this is 96 well plates where you put your Sanger reaction in each, so you can sequence 96 sequences at the same time. That looks ridiculous compared to what we can do nowadays. And this is a capillaries. So there's some needles that goes into the wells. And each capillary, you will have your DNA oligos, your fragments that are labeled with different colors, migrating along each capillary. And they end with a color, right? A color for A, a color for C, G, or T. And the speed at which they're going to reach the sensor is correlated to the length of the fragment. So you know exactly where this base is in the sequence. So, oh yeah, so here we go. It's hard. So I can pass this around. I think it's not only like cool, but it's beautiful in my sense. So that was just like to start to put some context, because now we can sequence millions of sequences at the same time. And with PacBio and Nanopore, even longer sequences of Sanger. So this is pretty impressive. So Giulio mentioned the Moore's law, and as the Moore's law for sequencing goes down faster. I mean, the curve for sequencing goes faster than the Moore's law. To give you some context here, in 2012 compared to 2022, we reduce the cost of sequencing by 10 times, tenfold differences. You may say it's tenfold, that's not that much, right? let's put in some context here. If you have a laptop that's $3,000, tenfold is $300 a laptop. A flight to Paris will be $80, and a nice dinner, $10. So to give you tenfold in science, it's a huge deal. All right, so I'm going to go through the basic of next-gen sequencing. As you described, next-gen sequencing is second generation and third generation. So they all look about the same. So you start with a DNA fragment. You're going to fragment a DNA fragment. Yeah, you're going to fragment it in smaller pieces that you can handle. Then you're going to ligate adapters. These adapters are going to be very important for the sequencing part, but also to insert these fragments onto a substrate. Then you do size selection because you don't want the smaller pieces, you want the one that are bigger. And then you amplify these fragments because you want to have enough fragments that are similar to have a bigger signal. After that, you deposit or you load on a substrate here. That's an example of Illumina sequencing that is covered with oligos complementary, the red complementary to the blue. And then you're going to amplify the template. And this is very important for next-gen sequencing, second generation, so the signal is strong enough that you can visualize it. So you create these clusters or colonies or polonies, depending on the type of substrate, that are identical sequences from the same original library. So now you can have 4 different sequences generated from each colony. And here you see the sequencing happen from 3' to 5' on each of them. So this is a basic workflow of next-gen sequencing. But this is general. This is true for second generation. In third generation, it's more simplified. Third generation, you don't have to fragment, right? You can use a longer. Some protocol you fragment, some others you don't have to. Like oxonanopore, like if you want to do ultra long, you don't need to fragment your DNA. But you don't have to amplify. You can if you don't have enough DNA, but you don't have to amplify at all. So amplify the library, but also you don't amplify the template. So that's why we call it single molecule sequencing. So no template amplification, no library amplification. So the amplification. So I will not go through library amplification because that's the same for everything. Template amplification, there are several methods to do it that has been done in the past. The first one created by 454, Roche, that also available on ion torrent and used to be on the solid, which is a dead technology now. But it's pretty basic. You have these beads that cover with oligos that are complementary to your library, right? So the idea is to be able to put one fragment on this bead and to amplify the fragment all around the bead, right? So now from one bead, you have the same fragment around. So to do that, you do an emulsion like a salad dressing. You mix oil and some reagents and you shake it. I swear you shake it, really. I mean, the original technology, you really have to shake manually. They came with a machine after that, but you shake at a certain speed and you will get, most of the time, 1 bead per little bubble. And this bubble the bubble will act as a macro PCR. So the fragment with a 1 bead, 1 fragment will start to colonize all around this bead. So now you have this amplify sequence that you can load onto your instrument. That was a very clever approach, but not very efficient in my opinion. Then you have the solid phase bridge amplification. That's what Julia was describing with Illumina. Now you don't have any beads, no emulsion. You just load it on the substrate. In the case of Illumina, that's a glass. That's the glass slide. So this is the 2D from Illumina. Sure. These slides are covered with various oligos complementary to the sequence. Same thing as described before. Your library is binding onto the substrate, and then you start to do the bridging. The way the bridging does is, as it's called bridging, is the library will band, ligate on the next closest oligo, and amplify and keep going, and you get this nice very circle. So all Illumina sequencers use this technology. So I can pass this around. So this is the latest NovaSeq, and it was nice. On top of that, the latest Illumina sequencers have whales on their slides, right? And you can see if you take the glass one and you tilt it, you will see this rainbow reflection. It's all the pattern flow cell. It's a bunch of whales. The whales allows you to load with very high density and still keep the clusters separate. And then you have something very clever, and that's something that was created by Complete Genomics and then purchased by BGI for their BGI sequencers. That's the nanoball generation, the in-solution DNA nanoball generation. So essentially, you have a library, you circularize the library, and then after you're going to ligate it together into 4 elements in one plasmid, and this will be amplified. So what you end up to have is this DNA nanoball that is repetition of the same constructs. And these nanoballs are deposited onto a pattern flow cell, and sequencing happens again. So this is all BGI sequencers have this type of amplification. Oops. All right, so the sequencing itself. I'm going to talk about some sequencing that don't exist anymore, but that give you some idea about exciting where we're coming from with the new technologies. Roche 454 pyrosequencing. Roche appeared in 2010. They have a working product, and they use this technique called pyrosequencing. Pyro is light or fire. So you sequence with light or fire. And the way it works is you have this emulsion again. You have this bead covered with all this identical library, and you're going to put it into a well, 1 bead per well, right? And then you start the pyrosequencing reaction, which is essentially luciferase. So what you do, you take you plate, and the plate looks like this one. There is 1 wells in that, and you throw some A's, A nucleotides. And every time the A is incorporated in the sequence, the well is going to flash with the luciferase. You wash it, you put some C's. Every time it's incorporated, it's flashing. So you know exactly the order of each of them. Now, the problem is if you have multiple A's, right? So the intensity, as you can see here, the intensity will be a little bit higher. So 2 A's will be twice the intensity of the flash of the light, or the length of the light. After a certain point, you don't know how many A's you have, right? After 20 A's, you don't know if you have 20 or 15. So pyrosequencing, and I can pass around just a piece of glass. But that was great. That was great sequencing compared to Illumina at the time, not for the throughput, but for the read length. You could reach 1 KB sequences. So very similar to Sanger sequencing, and very high quality too. So it's a dead technology. Yeah. Then ion torrent sequencing with pH. So ion torrents, I think, still exist. I Googled it this morning, and they still sell it. I don't know where.
It's,
in Europe, probably. What?
So I also used to present this saying, oh, it's an outdated technology, but then someone in the group said, no, I'm still using it. So like,
yeah, there is still some, and I think they changed the look of it, and it's particular niche. But the idea is, let's use what the polymerase does. The polymerase, every time it incorporates 1 nucleotide, is going to release a proton. You cleave H plus. So you could measure that, and that's what we call sequencing by pH, right? It's like measuring pH. You measure how many protons you have. So the way it works, the invention really has been this. So this is a semiconductor technology. There's millions of wells on that, and under each well, you have a sensor, an ion sensor. So you will, very similar to 454, but instead of being light, is going to detect H+, right, the proton. So you do something, you throw some A's, and every time you have a peak in H+, that means the A was incorporated. You wash it, throw some C, and you keep going like that. So he has exactly the same program than for 5-fold, right? Homopolymers are more complicated to sequence. So here, that's a graphic to explain exactly. So you incorporate, release a proton, and you can measure intensity of the proton levels. And if you have 2 T's in a row, intensity is double. Another way to look at it, I mean, it's the same thing. So it was pretty good at the time. Actually, it was very competing against Illumina, and it was equivalent to Illumina at the time. Now, Solid, another dead technology, and this one I'm sure is dead. Because I'm pretty sure I was the last one when I was at Duke. It's sequencing by ligation. And what they claim is that you're going to see how it works. It was the most accurate sequencer out there, even more accurate than Illumina. And the way they did it was pretty clever. Now, instead of doing synthesis, like meaning you incorporate and you measure the incorporation by light or dye, or this time you're going to ligate a complementary sequence to it. So the sequencing reaction would be this 6 base pair degenerate oligo with 2 base pairs, right? So it's going to bind, and you have a signal every time it binds. And you keep going, you ligate 10 times in a row. And you can, and with the color, you can know exactly which one it was, which duo it was. It was, if it's red, it's a GC, if it's blue, it's a GT, if it's AA, it's a yellow, etc., etc. So you will say, yeah, but how do you know which one is which, right? Because you only have 4 colors. What you do then, you unligate everything and do it again, but you switch by 2 base pairs, and you do it again. So each base is going to be assessed twice, right? So here you create an offset and you do it again. You create an offset, you do it again, and you keep going like that. So that was a very, very clever, but a very finicky technology, as you can imagine. BGI sequencing, sequencing by ligation as well. And that was again created by Complete Genomics. So they use these nanoballs. Nanoballs get loaded onto a pardon flow cell. And then you use the same idea of ligation. I'm not going too much into details here, but you're going to flank it. So you're going to sequence one on the left, one on the right, and ligate again, and do it the same way. So very accurate as well. Okay, Illumina. Illumina is probably most of you, so the stats of the survey know about Illumina, so we'll go fast. Illumina SBS, sequencing by synthesis. Reversible terminators, meaning that this time you stop, right? So you incorporate nucleotide, you stop. You incorporate another nucleotide, you stop. And each incorporation will have different color, right? If you incorporate A, it's going to be blue, the C is red, etc., etc. So you have a very finite read length. The other technology I showed you before has a variable read length, right? Because, yeah. So, so Illumina approaches Sodexta 98, and again, bridging amplification. And the sequencing, as I was saying, you incorporate a nucleotide of different colors every time. And you cleave the termination, and you incorporate another one. You cleave the termination, incorporate another one. So all you end up to have is this cartoon on the bottom of your page where you see these colors blinking. And actually you see that on the screen. Blinking for each incorporation, each cycle, they call it. As I was saying, there is this part of the flow cell. So now it's not just a piece of glass, it's a bunch of wells in it. So it allows you to have a higher density. So because the problem of the previous Illumina is if you have too high density, the cluster will touch each other, and it's hard to determine which cluster is which. With a well, the part of the flow cell, now you can really identify which cluster, and you can have more compressed in space. But Illumina also has other sequencers, the Nexus sequencer, where they don't have 4 colors. They only have 2 channel colors. So essentially, they have 2 channels for each. If you see, if you put a red light, you see red. If you put green, you see nothing. It's a T, you see nothing on the red, and you see the green only. If it's A, you see both. And if it's G, you don't see any of them. So it's a cheaper sequencing. They found a way to make cheaper to sequence by reducing the cost of the reagents. There are some problems at the beginning about biases, but it's better now and it works very well. PacBio. So now we go into third generation sequencing. As you can recall, no amplification of the library and no amplification of the template. So this is a very busy slide, but essentially the PacBio invention is not the sequencing part. It's this piece of material, this zero-mode waveguide. Essentially, in these wells, when light refracts, is very located inside the well and very intense. So you can detect single molecule level incorporation. So this time is different. You don't stop the sequencing. You have these reagents floating around with some A, C, G, and T with different colors, and they're floating around randomly. So when it's floating around randomly, the camera doesn't see them. But as soon as it's getting incorporated in the well with the polymerase, the polymerase grab it for 1/10 of a millisecond. And then you see the light, right? So there is a beam and you see the color. Long enough that you see the color and keep going. So because you never stop, you can have very, very long sequences and extremely fast because, as I like to say, you can sequence at the speed of the polymerase, right? So you have this particular library they call Smart Bell library with 2 hairpins on each side. I will go into that later. And each well will have 1 polymerase bound onto this Smart Bell, and it will sequence in a circle. I'll show you. So the read length is what we're talking about. Illumina will be on the KB, 250 on the 250 KB, 250 base pair, sorry. Oh, this is 40 KB, 50 KB long. Almost. So very, very processive type of sequencing. So this library has 2 Smart Bell, 2 adapters on each side. It's a linear library, but it's topologically circular. So what I mean is like the polymerase, the polymerase is going to be displaced, the strand, and generate the other strand. And now you can see this hairpin makes library circular, and you keep going. You keep going until the polymerase dies. Right. And this is a very important thing, and we'll talk about later about how PacBio improve the technology later on. I will skip this one. So PacBio is very good compared to Illumina and other technology, but GC bias, as Julio mentioned. So this is a pretty old slide from 2015. And you can see Illumina. So the x-axis GC content and the y-axis the coverage, right? So Illumina has a certain bias, like very, very low GC, very high GC are not very well covered. But PacBio that has no amplification, so no GC bias has been added, you can see it's pretty much the same, right? You get about 35 to 40x coverage across all different GC levels. The problem of PacBio is because you have no amplification of the template. That means you're sequencing 1 molecule at a time, which means that you see every errors made by the polymerase, but also errors with the visualization, with the capturing the information. So it's not buffered by a cluster like in Illumina or a colony. So this is how Illumina data, PacBio data looks like, the raw data. So it's very nasty, right? So you can see here, all the with a line and in purple is all indels. But the advantage is this error is random. You will talk about Poisson distribution. With Illumina, the error is random. So meaning that more coverage you have, with more coverage, you can improve the accuracy, right? It's not always the case for technology with biases, right? Illumina, you can increase the coverage, but the bias is still there, right? So it's very important for genome assembly that coverage is your friend. Coverage will be able to cancel these errors and reach something that's very similar to Illumina quality at the end. And of course, I cannot talk about long reads without talking about oxonanopore, right? And I'm sure a lot of you may have this at the lab. So nanopore is essentially, you don't synthesize, you don't measure anything but a flow into a pore. So this pore has ion flow going through, and every time the flow is disrupted by a sequence, you will have a signal that you can capture. That's as simple as that. And each nucleotide will have different size, different conformation. We distribute the signal. Yeah, 20 minutes. I'm almost done. It distributes the signal, and then you can know which sequence. So here another cartoon about it. So you have this flow going inside in the membrane and ionic current that is stable. Every time you have something going through the channel, you can capture the change of signal. So this is another animated version of it. So again, you're sequencing at the speed of the pore here. You don't stop. So it's very processive and you get extremely long reads. With some of the protocols, with no fragmentation, you can get up to 1 megabase, 2 megabases reads. To give you an idea, a bacterial genome is 1 to 2 megabases. So it's pretty impressive technology. And it's a technology that is very portable. So I would like to finish on long-range technologies. Long-range technologies are very important to be able to scaffold, and you will see that later in the workshop, scaffold the genomes. So the most important long-range technology we're using is IC. IC is chromatin interaction method. So here is a slide from Arima Genomics, one of the main provider for IC kits. So you're crossing DNA in the native form, you fragment it, and after you ligate adapters to it. And what you're going to sequence is with short reads is the junction of these two fragments that interact with each other, right? So this gives you a 3D information about how the genome was packaged. So you will tell me, how do you do that for assembly? Well, it's most likely that these two fragments interact if they're close to each other physically, right? If they're far away, they will not interact, they interact less. They may, like trans interaction, but it's more likely to be cis interaction. And this is what we're using here. So here is IC plot. When you see the interaction level, in the diagonal represents the cis interaction, and further away here is like the trans interaction. So here a cartoon to illustrate what I was saying. You will have 3 types of data. You will have long cis, is like these 2 reads you're going to sequence are connected with at least 5 kb distance or 50 kb distance, or you have short cis, or you have trans. And you can see here on the left, the bar graph, the different proportions. In gray is the trans, orange is the short cis, and blue is the long cis. So it makes sense. When you're long, it's easier to form. And so this, and what we want for genome sequencing scaffolding is the blue. So it's excellent for that. IC is of course used for other purpose as well, to study genome interactions. And I will finish with link reads. Link reads, it's give you the information, long distance information with sequences, but with gaps, right? So the idea is you're going to barcode every short reads of a longer fragments with identical barcodes, right? So after when you sequence this, you can pull them together, right? So for instance here, these 3 reads will have the same barcode. So after that, mathematically, you can separate these reads and assemble them separately. They should be together. I don't have to go into details about this approach. 10x used to have this discontinued product, and now you have other products by MGI and other TELSIC. They have various versions of the same approach. I would like to finish with binary optical mapping. That's not sequencing, but this is very, very useful to not only to scaffold the genome because it has a long range, but also to validate certain contigs or other scaffold made by other technologies. Because we call that orthogonal technology. We have biases, but different type of biases. So the way it works, you have this gigantic piece of DNA. Oops, sorry. That you're going to linearize. And through these pillars, it's going to become linear and migrate through these nanochannels. in each channel, one fragment will migrate at the same time. And then you take pictures. Actually, you take a movie here. So it's like a big microscope. What type of movie you're taking? Well, you're going to label this DNA. So each fragments are going to be labeled with a dye that will indicate restriction sites. So this is the way it looks on the screen when you see this DNA migrating through the nanochannels. So here you see in green is the backbone DNA, and in red are the labels of restriction sites. So now you have this kind of barcode, physical barcode of the DNA that you can align to a reference or align with each other. This is an example here when you want to scaffold contigs. You're going to align these restriction sites on each coding region, on each contig, sorry. And then you know that these two contigs go together in addition to that, because you're taking a movie of these DNA fragments, you know the exact size. So you can size this gap. You know exactly how much data is in this gap. So this is just to visualize how it will look like. On the top, you have a binano map. And after you have your contigs, then you can align. And in this particular case, it's 2 enzymes. Binano could be used also for other purpose. You can discover deletions in the genome, insertions, copy number variation. You see this particular pattern when you map to the reference that these restriction sites will be at different position, inverted or translocated. So it's very powerful and very complementary to sequencing technology. And I'm done. Any questions? I'm going to pass the one. This is Bioinformatics. That's what it looks like. So as 3 full cells, and you don't see the nanochannels.
I'm gonna check the chat.
Yeah.
Okay. I'm gonna quickly just turn this.
Oh, no, there is one.
Get them before I have to show one of the chips.
There is one question in the chat.
Yeah.
Are there biases that are species specific, or are they mostly dependent on the sequencing technology?
Yeah, I mean, some sequencing technology will behave differently with different species. For instance, on nanopore, there was this issue with secondary structures. So if your DNA goes through a pore and create a secondary structure at the end, you will block the pore. So you have this sequence underrepresented in your final data set. And we noticed that in some birds, in chicken or in fish, this happens more frequently than other species. So yes, some biases are more prominent with certain taxa than others because of their underlying
Characteristics
of the genomes.
Okay, great. Thank you, Olivier. Other questions here in the room?
Yeah.
The bio coverage is depending both on how many molecules you get between the barbells, but then also how many times you go around the circle.
Yeah. So I will go after later with I-Fi and the difference between what they call the continuous long read CLR and I-Fi. For the CLR, you can get 40, 50 kb, very low quality. But then with what you're mentioning is the going around to have a nice consensus. But yes, it will depend on the insert length. And I will show later on about 20 kb insert length, and you go around about 10, 12 times.
Yeah, so we will dive into this later. Just a few housekeeping. Basically, now we are going to set up everything for the practical, and we have Linnell who is going to is our expert on how to use Galaxy, and she's gonna basically guide us through the whole thing today and tomorrow. However, because we have many people online, there is also Nadolina who is on the call. So we have a breakout room, and if you have any problem, please reach out to her in the chat, in the Zoom chat. Then you can join Nadolina in the breakout room, so that basically, you can solve directly with her any problem you may have, setting up your account, running jobs, anything. Okay. Likewise, here in the room, as we go through a practical, maybe there will be no problems, but if there are, we have Cassidy Johnson. She's a graduate student here at Rockefeller. She has also experience with running things in Galaxy, so we'll be here with me as well. And if you have any problem, just, I don't know if we have to post the thing or.
I'll explain it when we get up to that.
Okay. So basically, you can ask us for any problem you may have.
All right, so as Julio said, this is just gonna be a quick little section, just like a little housekeeping one. So I know that Matt sent out instructions for signing up to our VGL Galaxy instance, and I know at least like 15 of you have followed that. So if you haven't, I'm giving you the chance now to do that while I give a general introduction to what Galaxy actually is. So for anyone that has, this is what it looks like, the webpage I'm telling you guys to sign up on. And I'll have a URL here. So you need to be on either VPN if you're not on campus, or the RU Wireless Secure Campus Wi-Fi if you are on campus. And if you should be able to access this URL, vglgalaxy.rockefeller.edu/8080. And that'll take you to, it'll look like this since you don't have an account yet. And you'll be able to register there. And for participants who have already registered, please log in so that we're ready to go for our practical sessions in a bit. If you're having trouble, reach out in chat, but generally the main culprit for this not working would be if you're using any extension that makes it HTTPS everywhere, you'll need to either disable that, or if it's not working, use an incognito or a private window, because that'll disable anything and just have the site fed as HTTP, which is how it works. So as a general introduction to what Galaxy actually is, it's this computational infrastructure. Yeah, I'm not doing anything here. I'm just introducing Galaxy. Yeah, but yes, sign up now while I'm talking or log in. So Galaxy is a computational infrastructure that helps you run a lot of bioinformatics workflows. It's one of the main selling points is that it's free and offers a lot of compute to people who do not happen to have that in your labs. There's 3 major servers, worldwide servers. There's the EU instance, there's the main instance for the United States, and Australia is working on setting up theirs. And this one that isn't the main one is the one that we use. It's called the VGL Galaxy instance. So it's running on our cluster. We've set it up to run all of our workflows using our compute, because we have 28 nodes that we can take advantage of on the cluster. But prior to using this Galaxy workflow, that would look like a standard bioinformatics pipeline carried out on the command line, which would be typing on a text-based adventure screen on the whole entire assembly process. But so Galaxy now offers, Galaxy will offer a GUI, which is a lot more non-environmentally friendly, user-friendly for people approaching it who are new into bioinformatics, who haven't done any terminal work. So instead of having to maneuver just by a text, it'll have it set up in this sort of interface. You'll have what's called a history, where you're running all of your jobs. And so it shows you if your jobs are running. So these are various outputs of one job. They're yellow, which means that that job is currently in process. It can also queue up your job. So that's the gray one. This means perhaps it's waiting for these to finish. Like one of the inputs for this job is an output from here, or maybe it's waiting for resources. And then you can't see it here, but it'll turn green if it succeeds. Yeah, so this is the basic overview of the interface. And the other key advantage of Galaxy is it helps make workflows a lot more reproducible, because you can package them up in workflows. And this example is this is, for instance, our HiFiasm, I believe. This is our HiFiasm workflow. So you can see all the steps. And if you're familiar with command line work, it's as if you took like a bash script and made it into just a flowchart type thing, with the jobs leading to each other, inputs feeding in, outputs feeding as inputs to different jobs. And you can also select which outputs you want to keep or which ones you want to discard, which are temporary. So all of this is what would be going on under the hood. But when you actually run this workflow, what the user sees is just a simple input form. You just tell it which, for instance, which data you're using. This is which lineage, some of the parameters of any tools being used in that workflow. But for our workflow, we have a lot of them kind of hard-coded for vertebrate genomes. For instance, for Busco, it wants what evolutionary taxa you want to look at, and we just have vertebrata hard coded in, but you can always change that. And you can also just use whatever you want if you're, for instance, running it on your own. It has free text, so you can add in free text if that's necessary for your parameters. This helps you name a lot of stuff further down the line so you can keep track of the steps in your workflow. Because in the practicals, we'll be doing a lot of, so we'll be doing this, but step by step. We will not be running a workflow, we'll be running it step by step together so we can all get kind of a better grasp on why each step of the genome assembly workflow is in there. So it'll be a lot of, it's not a lot of manual work, but you will rename stuff manually, and so that's why I'm plugging this. Yes, and also another highlight of Galaxy is that a lot of the public servers have integration of also public repositories. For instance, our GenomeArk that Julio had mentioned earlier is linked up on Galaxy EU, and I believe should be on org at this point as well. So anyone can just import our data from GenomeArk, such as our raw reads, and just do an analysis there. And prior to setting up the VGL cluster instance of our Galaxy, I did all of the genome assemblies, like 20 of them on the EU instance. So it is like, it's usable. It's a usable, robust public service. Yeah, that was it for my intro. Does anyone have any trouble or questions with like signing up or anything like that? Let me see if I can pop the chat open. Let me see if I can. 2 questions in chat. What's this say? No questions in chat. Okay, I'm gonna presume that means everyone's good. Okay. Let's see. Hide this again. Hide.
Okay, so before we get started with the actual workflows and tools in Galaxy, let's review a little bit more general principles when it comes to bioinformatics. So we already mentioned before that 1979, sort of the original idea of shotgun sequencing. You fragment the DNA, find the overlaps, reconstruct the original sequence in a systematic way. So that's about the time that essentially computers were developed to a point that we have personal computers, and also in the 80s and 90s. This is a funny review paper where essentially they say, though, in the 80s and 90s there were a flourishing time not only for pop music but also for mathematics. And that's true, again, because of the essentially the increase in compute power. And that's also when we have the development of things like GenBank. We start to have a lot of sequences out there. We have to store them somewhere. So that's when the NIH develops GenBank. Currently is the largest publicly available database of sequence in the world. It's been constantly updated over the years. There are many different DNA sequence database, the gene, the nucleotide. You have the whole genome sequence data. Then you can have protein databases and annotations and so forth. So you're, of course, in GenBank, you also find what we're after here, that is the genome assembly, so the referent genomes. We can define a genome assembly as the entire genomic sequence derived through a de novo, so reference-free. So it means we're not guiding our assembly using a prior existing assembly for the same species. That's de novo. Verve sequencing reads, and it's been usually curated, so there is a manual process involved where someone has been looking at this de novo assembly that was generated by the algorithms, and then it's made public. And until now, really, we're talking about single reference individuals or pooled individual in a single reference. More recently, there has been this concept of pan-reference genomes that can pool multiple individuals and still show the variation, but we're not going to dive into this. It's usually an haploid genome. So even if we are all diploid human beings, vertebrates are all diploids, plants can be octoploid and so forth. The actual reference is a single representative haplotype, essentially, for a species. And actually, with all the sequencing strategies that Olivier talked about, so except the latest long reads, we were talking about squashed haplotypes. So essentially, you had a mixture of both paternal and maternal haplotypes combined into a single haploid representation, because To resolve the haplotypes, you need these long reads. And sometimes the alternative alleles were reported as variants. Okay, so yeah, sometimes there is more than one assembly as well for the same organism, of course. This is, for instance, the primary assembly for GRCH38, so the reference, the human reference that has been used for it basically is the descendant of the original draft in 2001. And again, the primary assembly is what gene bank curators consider the most up-to-date source of the genomic sequence of this particular species. So you get a few info on that in this summary page. I mentioned before this GRCH38, it's the reference that's been used for a long time. It's still used for most applications. However, it's still incomplete. So there is another reference that from it's a different cell line that was used called CHM13, which is complete. And if you're interested, because you're working on humans, I will encourage you to look into that because it will probably provide better results for many analysis. So when you look at the primary assembly, you have, for instance, this is again a summary page from NCBI. For instance, for this particular sequence, you have number of regions with alternate loci or patches 317. That's the total sequence length. You have still 350 gaps again in GRCH38. That's why again I'm talking about this other alternative assembly was generated and released this year. The scaffold number 472, again, this is telling us that it's fragmented. And then we have these two magic numbers here that when you're dealing with genome assembly, people often look at N50 and L50. There is one for scaffolds and one for contigs. So just as reference, as Olivier was showing before, the primary difference between contigs and scaffold is contig means a sequence uninterrupted with no gaps in it. Okay, then when you start to have gaps because for whatever reason you didn't sequence that region, you don't know exactly how to reconstruct the sequence of that region, you place a gap in it. No, and it's a run or a stretch of ends, essentially, usually in the sequence. Then you have a scaffold, no? Because you have contigs that are separated by gaps. Okay, so that's why there are these two designations, contigs and scaffolds. But and of course, you have the number of contigs that is always bigger than the number of scaffolds because again, the scaffolds are combinations of contigs separated by gaps. The total number of chromosomes, so including plasmid, is what you would expect. And so let's look a little bit about these two numbers. So the N50, can be defined as the sequence length of the shortest contig at 50% of the total genome length, which is one definition. I think it's very hard to understand, but I'm going to try to explain it. And then I have a little exercise that we can all do together. That will allow you to essentially develop your own little algorithm on how to compute that. So here we go. Imagine that we have, our very small simplified genome that is made of 7 contigs, let's say, or 7 sequences. The first sequence is 100 base pair. The second sequence is 70 base pair. The third one is 60, and so forth. And essentially, these are ordered by size already. So this is kind of informative of how you can compute this particular value. And again, the N50 is the sequence length of the shortest contig at 50% of the total genome length. So if we consider the total genome length here, which is 400 base pair, 100 plus 70 plus 60 and so forth, that's 400. Half of it would be 200. And so in practice, the N50 is the size of the contig in this case, that essentially when you start summing from the largest contig to the smallest one is where you stop. Now, because it's 100 plus 170, I haven't still reached 200. Then I add another 60 and then I have to stop because I'm beyond 200. And so basically these would be your N50. And the L50 is 3 in this case because you have to some 3 contigs to get there. So these values are used all the time. So whenever you're comparing genomes, whenever you're comparing row sequencing data, people use them, and it's just something that is now part of bioinformatics in its broad sense. So it's good to have an understanding of how this works. I have a couple of examples here, and if you were able to join the Slack channel, I put in the general Slack the channel that's called General. I put an Excel file with these numbers. So for case A, B, C, and D, if you can open it, I'll try to do the same. Maybe, I'm sure I can do that. Do you have I want to open the Excel that I put in the Slack channel for this exercise. Oh, let's see. Okay. So again, you have to develop a little algorithm. Of course, I mean, most algorithms can really be, made by hand. They don't have to be written in a programming language. And again, here, even with this very simple example, let's see who gets the N50 first. So we have example A, B, C, D. Again, you need to find a little algorithm that will allow you to compute the N50. And recall the N50 is essentially the size. Another way to phrase the N50 is everything that is all the genome, half of the genome is in pieces that are of that size or bigger. Okay, so imagine the N50 is 50. It means that all the half of the genome is in pieces that are 50 base pair or longer. Okay, so imagine that in this case we have 20, 10, 5, 15, and 2. Are people online seeing this too?
They should be. I'm sharing the whole screen.
Okay.
Double check.
Can you confirm you're seeing this online?
Oh, yeah,
Awesome, thanks. Okay, so we have 20, 10, 5, 15, and 2 base pairs. These are the fragments, actually kilo base pairs for this example. So the first thing, if you recall my example, is essentially sorting them by size. So if we sort them, we can start to compute. And again, sort is an algorithm, so that will be the first algorithm that we use. We sort by the largest to the smallest, okay? So that essentially mimics what I had before. Then again, we have to compute their sum. And also, we want to compute the N50, so 50% essentially of the genome, so we divide by 2. So that's 26. Now, half the genome size is 26. So again, we want to see how much of the genome essentially is in fragments that are bigger than a certain number. So here you can do, it like this, then you sum. I'm summing the previous cell because then essentially if I now apply the formula to everything, I start to have a cumulative sum. Okay, so the first is 20, then 20 plus 15 is 35, 35 plus 10 is 45, plus another 5, 50, and then 2. Okay, so now recall, basically you have to start summing and then you stop until as soon as you've reached the half the genome size, which is 26. So in this case, it means, well, when I get to sum 20, 0 plus 20, then I have to stop. I have to still add. So I add 15, and that becomes 35. So that's where I have to stop. So our N50 here would be 15, because that's the size of the element that is when you stop. So 35, that's 15. Does it make sense? You want to try the other few examples? Particularly C and D would be sort of revealing a little bit of why this is useful, but also why this is can introduce some bias in the analysis. So I'll leave you just a couple of minutes to try it out. What's that? For which one? For the first one? No, so the N50 for example A would be 15. So you start summing here. Oh, L50, yes, L50 would be 2. Yeah, exactly, because you have 2 components. Yeah, sorry. I thought you were referring to this 2 here. Okay, yes, exactly. Okay, so yeah, so in the meantime, you can also start thinking about what could be a limitation of these statistics, because essentially this, of course, is a summary statistic. So the idea is that you use it to infer some properties now of a combined set of elements. Now here you may have millions of reads, or you may have hundreds of contigs. And for some reason, and maybe we can think about that together, people decided not to use the average, but use this fairly more complicated to compute metric to try and understand and get the sense. And again, it's so popular that if you go on the primary assembly page for GRCH38, the human reference, that's the only metric that you get. Now you don't see the average there. And there is a reason to that. If we try now to solve examples C and D, we can see why is that. So again, we have to, well, first we have to order them by size. Okay, sort by size, largest to smallest, then we sum everything and divide by 2. And then again, this is this cell plus this cell, cumulative sum. And again, here, so we start 130 is less than 257, so we're good. 250 also less than 257. Oh, now we have to stop here. So basically our N50 in this example is 100. And the L50 would be 3, right? Okay, so if we do the same thing here, let's see what happens. I'll do the sum first, but then I will still, sort them by size. Okay, so of course the sum, I mean, this is an example. So the sum is the same as the previous example. And even in this case, the N50 is still equal to 100. But what's the difference between example C and D, even if you just look at it here in the slides, no?
C is more contiguous in general.
Right, there are fewer pieces, right? And the reason for that, and that's also the reason why it was designed in the first place and became so popular, is because essentially everything that is below the 50% doesn't count at all. It could be, a single base pair fragments, doesn't matter, because it's completely not important how you compute this. Only the first 50% in terms of size of the genome really counts, or whatever you're measuring. So, for instance, when we're thinking about long reads, for a long time, long reads, yes, they're long, but they also produce very short fragments sometimes, either because the original DNA is highly degraded, and so there are a lot of small fragments that were not removed during the library preparation steps, or for any other reason. Essentially, you have a lot of long reads and a significant amount of very short reads that you want to disregard entirely. And this kind of metrics essentially disregard them entirely, because it doesn't matter how many small fragments you have, you just don't count them at all. Whereas if you were to use the average, if you have small fragments of one base pair and you start to do the average of all your sequencing, basically you get very, very poor results, and you start to get confused about what is exactly the status of your library. So the N50 works for everything from sequencing reads to assemblies. And again, it's just a way to say, well, most of the stuff is in big chunks, let's say, or chunks of this size. And you don't really consider the small parts. But of course, the drawback of this is that because you're not considering any of the small parts, there could be significant differences when you compare two things and you just don't observe. Okay. All right. So can I? Yeah, I can. Sure.
So if the you recently said bigger than the counting.
Well, the scaffold.
Yeah, scaffold. If you ignore the gaps in scaffold, why you still got so many scaffolds? Why if you can ignore?
What do you mean ignore them?
I mean you said there's no gap in the counting.
Yes.
And, if you have the gap in the scaffold, why can you just get a one nice scaffold?
So if you can still ignore them, you mean? So the gaps in the scaffolds, they represent a known sequence, but because the overall, the overarching goal is to reconstruct the entire chromosomes, we allow gaps, but at least we know the order and orientation of the sequences within the larger chromosome, even if we don't know exactly the entire sequence. Does that make sense? Okay. Yeah, so let me start sharing again from the whole the presentation. Just a second. I don't know if there.
Just share the desktop. I will just share the desktop.
Oh, really? Okay, sorry. So that's it. All right. So a few more things. We have again, we have a gene bank, but there is a counterpart in Europe, the Ensemble Genome Browser, launched in 1999 by the Wellcome Sanger Institute and the European Bioinformatics Institute. That was essentially used to store initially the Human Genome Project data, but then was used for everything like GeneBank. We also have the UCSC Genome Browser, started in 2000. It can visualize and download genomic information on many vertebrate and invertebrate species. It has a lot of features that are similar to GeneBank and Ensemble, but it's generally a bit more curated, but also less species available. Let's talk about some stuff that maybe is already known, but I think it's important. File formats. FASTA. It's the most popular and oldest file format to store sequence data. So in 1985, it's pretty basic. You have what's called a header line on top, with this symbol that starts the header line, and then you have the sequence. You can have as many sequences as you want. And essentially, this is how we store our final assembly files. We have FASTQ formats, a bit more complicated. Is essentially what Illumina generates, but also other sequencing platforms. And usually it's used for reads. Essentially, you have another header line, which is a bit more complicated because it gives you a lot of information about the sequencing platform that was used to generate it. Then you have the sequencing, the sequence of that particular read. You have a sort of a separator, and then you have the actual quality for that particular read. So essentially, there is a matched quality for each base in the sequencing read. And again, this is inferred by essentially the quality of the signal when the sequencing was performed. Okay, then, yeah, this is how you describe all this stuff on top. If you're interested now, you can really get a perfect, it can tell you the lane, the tile, the X and Y position, and so forth in the flow cell. How you read the quality, that's important. It's based on this ASCII code, okay, where essentially the logic is that you need to be able to assign a number that corresponds to the Phred score, the quality score that I mentioned before, to each base. Okay, so it's a numerical evaluation, but because numbers are usually more than one digit, that's a problem because you want to have the same number of characters in the sequence and the same as the same number of characters in the quality. So you use the ASCII codes essentially, to give to assign to each character to a number, and then you get the original sequence quality. The genome assembly problem, is often described as essentially find reliable overlaps. Okay, so if you find all the reliable overlaps in your sequencing data, essentially you can reconstruct the original assembly. So again, in this little introduction, then we're going to dive a little bit more into genome assembly algorithms. But as a general introduction, let's talk a little bit about alignment of sequence data. And it can be reads, or it could be, if you're doing comparative genomics, is how you align two different sequences, or from, let's say, different species to each other. And essentially, you can use it for a lot of things in bioinformatics. No, there are several ways. Now, there are like hundreds of algorithms for alignment, but there's general several ways of doing alignment. Essentially, we started with what's called pairwise alignment. So you only have two sequences and you want to align them to each other. This can be generalized to multiple sequence alignment. There is a specific type of alignment, which is read mapping. Now, when you have millions of, for instance, Illumina reads and you want to align them to a reference genome, so it means that you have one single reference sequence and millions of reads that have to be independently aligned to it. And then, of course, you can even scale this up to aligning all genomes and so forth. So how the general principle works, you have a few things that are necessary for an alignment, usually. You need a number, the so-called quality score that defines the quality. Now, because you can always create, an alignment between two sequences, even if they have nothing in common. You can always try to do that. No? The point is that you have to be able to assign a quality score to that alignment that represents the likelihood of that particular alignment to represent a real biological similarity, essentially the alignment between two regions of the same, two sequences coming from the same region of the genome. So in order to get to that quality score, you need what's called a scoring scheme, through which essentially you can determine the quality score. And to get the scoring scheme, you need what's called essentially, to determine what's going on here. Not touching it. I don't know. It's failing us. Sorry, for those online, we have a bit of a problem here in the room. We'll resume in a second. Okay, thanks. Should I try to disconnect or reconnect, or
Sure.
We could take a break now while we fix it.
That's an option.
maybe I just, okay, I'll try to continue now. Okay, so yeah, the alignment score is the sum of all the substitution and gap penalties. So we have two things essentially that defines our scoring scheme. Usually you have substitution penalties, if you have a difference of one base, and then you have gap penalties if there is something that just doesn't align and you want to open a gap between two sequences. Usually you represent substitution scores like matrices, like in this case. So of course, if you have, in one sequence you have A and the other sequence you have A, then you give it a score of 1, so it's a match. Whereas if it was a C or a G or a T in the other sequence, that you get, for instance, -1. And of course, this can vary now because there are some, for instance, transversions or transitions that have potentially different substitution scores, but you need some kind of metrics. And so then how do you in practice generate the alignment? Okay, so brute force is sort of obvious solution that never works in informatics. But the idea is, okay, well, let's try to generate all possible alignments, compute the quality scores for each one of them, pick the alignment with the highest quality score, and we're done. Of course, that's not feasible because if you have a lot of even short sequences. Now, if you take an example of 100 base pair sequences that you're trying to align to each other, you have an astronomical number of potential alignments. So there is, even with the power of a CPU, there is no way you can do that. So they, again, in the 70s, they started to develop algorithms for that. The most popular and still used one is this Needleman and Wunsch algorithm in the 70s. And basically it gives you the best solution, so the best optimal alignment between two particular sequences, of course, given the score scheme and so forth. It's referred as optimal matching algorithm as well. It's kind of approach. It takes the canonical approach in computer science of dividing a large problem into smaller problems. It tends to maximize similarity. Goes from the smaller problems to repetition uses the optimal global alignment. Is uses for optimal global alignment. So it means if you have two large sequences and you want to find their optimal global alignment, not necessarily the best potential alignment of sub-regions of the genome of the sequences, that's what you want to go for. In general, works well when you have highly similar sequences, but it gets very, very slow as the length of the sequences grows. So I will just quickly show here the principle. Again, you have a matrix. It's not a matrix like the scoring scheme before, but it takes advantage of it. Again, imagine we have two sequences that we want to align, the one on top here and this one on the left. Usually you put a 0 here on top, and then basically you have to assign certain numbers based on a few criteria. I'm not going to describe in detail how this works, but if you just go on Wikipedia, you can find out how this works. But essentially, the neighboring cells, that's why it's a smaller problem that sort of you solve and then you move to the larger problem. The neighboring cells inform you how to populate the matrix, not like in this case here. And so you start to, you ask the question, what number should I put here? I will look into the other numbers. And when I have all my matrix populated like that, then essentially the matrix itself is revealing of how the alignment should be performed. Because essentially by looking at the numbers, of course the negative numbers tend to tell us no, this is not a good alignment. So we start from this position here and walk our way back to the origin of the sequence, and we can define all possible paths in the matrix that have sort of low values and define the potential best alignments. And there is of course necessarily not a single alignment available. Other general concept that is actually has been demonstrated to be identical to this. It's this Levenshtein distance or edit distance. It's another way to compute the sequence similarity between sequences and find ultimately potential alignment. So here you can see, if we have two words, this banana, which means the same in English as in Italian, and bambino, which means child in Italian. You can go from one word to the other by making a series of substitutions and insertions or deletions. So in this case, now you can add an N. So you can substitute an N for an M and get from banana to banana and so forth. And then you get to this final word. So there have been three changes, 2 substitutions and 1 insertion. So the edit distance is 3. And again, this is also another way to find alignments. Just to mention that this approach that I described before has been generalized for what's called local alignments by Smith and Waterman in 1981. Works essentially can compare segments of all possible lengths, so it's not like bound to have a sequence of very similar length. And again, it will find the optimal local alignments, the local best solutions. So again, it works in the same way. There's this matrix. You have to put the numbers in the matrix according to the rules, and then you can find all the possible ways to walk through the matrix and find the alignment. Of course, then you can have tons of approaches that have been combined in mixed global and local alignments. And then there is something that will be important for the next session, It's called usually reference-free alignments, even if it's a little bit misleading because there is not necessarily an alignment involved in the overall theory, but this is probably one of the most important concepts in mathematics today. So essentially you can quantify sequence similarity without essentially aligning the sequences or in what's called a pseudo alignment. So the reason why you may want to do that is because, for instance, if you're doing a genome assembly, you want to find all the possible overlaps between reads, but because computing the alignments with the approach that I presented before takes a lot of time, now you have to do a lot of math. Essentially, that's very slow. And if you have millions of reads and you want to try to align all the reads versus all the reads, then it takes extremely, it can take a lot of resources, whereas pseudo alignments will give you sort of the first intuition as to which reads should be aligned to which other reads, and then you can focus on these alignments only. There are other use cases, for instance, when sequence collinearity is violated. So when you have, for instance, recombination, transposable elements, any other things that essentially violate collinearity, the approach that I presented before will struggle a lot, whereas these reference-free alignments will not. Or where you have very low sequence similarity, and so the numbers in the matrix don't really make sense. This is also potentially effective. Or if you have, again, like in the case of genome assembly, essentially there is too many alignments to compute, and you cannot just do this kind of attempt with, for instance, Needleman and Wunsch for each one of them. And the other thing that is good is it's usually assumption-free, so there is no scoring matrix involved. It's much simpler, and it's based on this principle of words or k-mers. Do you know what k-mers are? Okay, so we'll dive into this. So imagine we have two sequences, and again, we want to find their similarities. We have this pseudo alignment. So we have sequence X and sequence Y. Okay, we can kind of see that there is some similarity, but it's not clear how much similarity we have. So with the k-mers, that is the same as a word, and the K stands for the length of the word. So imagine we have a word size of 3 in this case, or 3 mers, or k-mers of length 3. Okay, we can dissect the two sequences that we want to compare in all their constituent k-mers. So we have, in the case of the first sequence, we have ATG, and then we do this in sliding windows. So we move one nucleotide to the right. So the next k-mer is going to be TGT. Then we have GTG, TGT, and GTG again, right? So that's for the first sequence. We can do the same for the second sequence, and we get CAT, then ATG, TGT, and GTG. So now that we have dissected the sequences in k-mers, essentially we can compare the k-mers instead of comparing the original sequences. So in this case, we can check the union of the two sets. Okay, so the union of the two sets means take all the k-mers that we have ever seen. We have CAT from the Y, ATG from both sequences, TGT from both sequences as well as GTG that is also present in both sequences. So there are some k-mers that are shared and some others that are unique, CAT in this case. So we then can count the words or count the k-mers. So we can see that, for instance, the k-mer in X as 0 counts for CAT, 1 for ATG, 2 for TGT, and 2 also for GTG. And then the other sequence has 1, 1, 1, 1. Then there is something called Euclidean distance. So you can essentially take this as a vector for sequence 1 and sequence 2, compute the Euclidean distance of these two vectors, which is done by this formula. And you get a number that is this Euclidean distance. So this is a number that doesn't mean anything particular if you're only computing the distance between two sequences. But if you have millions of sequences, you can compute this Euclidean distance for all of them. And you get the ones that essentially have a lot of similarity. We have, the Euclidean distance will tell you, how close they are to each other. And then you can use this information to say, well, there is no point in me trying to align this particular sequence with that one because they have no sequence similarity whatsoever. I will just focus on the ones that are more relevant. Yeah, so this has been used, this approach with k-mers. If you go back in the 1970s, they were already using k-mers, even if they were computing them by hand. And it's extremely fast. It's been used for transcript quantification, SNP calling, metagenomics, barcoding, you name it. And of course, genome assembly. BLAST, now I'm sure everybody has been using BLAST in their research. It's based on this principle, you have query sequences, you dissect them into words, find essentially the matches of the k-mers, and then try to extend these matches between the sequences. And it's been out there for 15, almost 40 years now. So it's it's a very effective way. And in fact, I mean, the reason why essentially this is also useful for genome assembly is because, especially when we were dealing with short reads that were like 100 base pair long, you had a problem now because essentially, if you were looking at a 100 base pair long read, basically you didn't have, you didn't have a lot of flexibility because if you were to break the sequence into its constituent k-mers, then you could find those k-mers in other reads. But if you were to try to find overlaps directly between the reads, what you would end up with is essentially that if there is even a single error between one read and the other, essentially then you will not be able to find a perfect overlap. Whereas with the k-mers, you could avoid that. So that's why essentially that they have been very popular, and all the assemblers, or most of the assemblers in the sort of pre-third generation sequencing era were based on k-mers, and this concept that we're going to discuss later on of the Bruin graphs that are based on k-mers. Okay, so with that, I think we are at the break, or no, we first after this. Okay, so we start the first practical session using k-mers to determine the properties of the genome without actually assembling it. And I was gonna guide us through this.
Just a quick note, so if you'll notice there's a what?
Yeah, you have to come over here.
Oh, they can't.
Yeah, I don't know if they.
For the people in the room, I have Post-it notes at most of the tables. If not, just grab them from another one. This is kind of just like how we're gonna try and manage who needs help without having it be disruptive. So it's a green, blue, and an orange Post-it. If you are stuck and like actively need help, just put an orange Post-it on your laptop, and then Cassidy or Julia will come around to help. If you're doing okay so far, you could put the light blue one. And if you're like green is like good to go, ready to move on to the next step, then the green Post-it will suffice. Okay.
And again, for those online, Nadalina is in the Zoom as well, so you can coordinate with her through the chat to use the breakout room anytime. Or if you have a problem, you can talk to her, okay? Or you can even try to talk to us.
I'm gonna real quick just fix up the sharing because I want to share half my screen. So let's try and figure that out. Okay. Maybe it's better. Okay, this is visible. Okay, that's fine. Desktop. Okay, and we're back. So I just have to split my screen real quick because I'm gonna have some of the practical, I'll be doing it alongside with you guys. But so that's why I have my own little galaxy signed in on the other side. But I have just a couple slides of introduction first before that. So I'll just make this again big format. Okay, so what this practical will be, what we'll be doing this practical is using this tool called Genome Scope 2 to get an estimate about our genome's characteristics based on just the raw reads. So you only have your hi-fi reads or Illumina or whatever reads you use. And it uses those k-mers that Julia was talking about to create a k-mer histogram. So it's coverage of various, it's the coverage of all the various k-mers found throughout your reads. And these distributions, it kind of looks like a coverage distribution, and you can use it to infer coverage, but it can be very informative as to a couple different characteristics of the genome. And it's also gonna change depending on how the genome actually looks. So I have just a couple quick examples that I'm gonna walk through. So yes, as I said, a k-mer spectrum is how often do these k-mers of your chosen K occur in these sequencing reads. And the spectrum represents the complexity of the genome. So haploid genomes, such as this little yeast, one of these yeasts here, has usually just one peak. And then a diploid genome will have a more obvious two peaks. This second peak is the one of diploid coverage. So this is k-mers covered at here 40x coverage. And this peak is the haploid peak. So it's k-mers covered at 20x coverage, or half of diploid coverage. So these would be the heterozygous portions of the genome, and these are usually the homozygous portions of the genome. The spectrum can also show you sequencing errors, which are k-mers of very low coverage, and they're going to be low coverage because they only occur, they're not actual real biology that's getting sequenced a bunch of times. It's an error in the sequencing, so it only occurs like a few, if any, mostly just one times. Mostly just once. And that'll be this to the left of this red line here. And it also shows, it can show repeats, which can create peaks or higher k-mer frequencies, or if you have an individual, if your specimen has aneuploidy higher than diploid, you'll see corresponding peaks here at a predictable coverage. So if you have a triploid, then your homozygous triploid would be at 60x, that peak here. And so this is what a very heterozygous diploid organism looks like. This is one of the zebra finches that we have. This one's mutated too. So for a diploid individual with really low heterozygosity, so remember that this peak is corresponding to the heterozygous portion of the genome. If you don't have a lot of regions that are heterozygous, then you're not having a lot of regions that are covered only at haploid coverage. So for a diploid individual that has low heterozygosity, such as this endangered rhino, this peak is going to be a lot smaller. It doesn't look as bimodal. It, if anything, looks like a little shoulder hump, but it's still there. And you'll see GenomeScope also provides some characteristics, just kind of spelled out in summary. It estimates your genome, the length, the size of your genome. That's the length up here. What portion is unique versus repeats. This is homozygosity and corresponding AB heterozygosity. See, it's 0.21% for the rhino, whereas it's 1.47% for the zebrafinch. This K-cov is that haploid coverage I was telling you about. It's calling the peak at 18.5, which is near the 20 that I mentioned earlier. Error percent, these k-mers, duplication, and K, these are just the parameters you ran it with. So you can run it with various types of K, but it has to be the number that you made the database with. We'll get into the database in a minute. Here it's 21, and then P is just diploidy because this is a diploid bird. Okay, so that's just an overall view of what GenomeScope does. This is the first step in our pipeline. We run this before the assembly, just so you know what to expect, and also it helps give us parameters that we'll use in later workflows. For instance, we use the genome length to cover it, to calculate NG50, which is like N50, except instead of summing up the sequences that you're giving it, you give it the expected genome size, which we're using here from GenomeScope. And we also use like these coverage estimates later on for things such as purge dupes, which we'll get into later on. Okay, so this is the sort of practical part. If everyone could just go onto their Galaxy and their browser, please. Let me split these screens again and hide these controls. Okay, let me see. Make this as big as possible. All right, this is good vision wise. Like you can see still the notes on the side. Still okay? Oh wait. So first thing we're going to do is we need to make one of those histories that I talked about earlier. So we can, when you start up Galaxy, you should be given this unnamed history automatically. So we're gonna go ahead and we can name it BGP workshop or whatever you want to. So to rename it, you'll click this little pencil icon. I'm just gonna call it VGP workshop. And then you'll save it. And so the first step to analyzing data is having data. So we'll go and get some shared data. This is going to be a yeast data set that we use for testing and for training. It's mostly, I think it's synthetic hi-fi, but the BioNano and Hi-C come from a real experiment. So go to shared data and navigate to data libraries. It might be slow because now we have, this is most people ever using it at once. Sorry, I'm not actually in IT, but so we're gonna click onto yeast example data. It's this yeast data set. Don't click on the Chernobyl one. That's not what we want. Yeast example data, and just click through a couple of these links until you get to a page that looks like this. So we're going to have one BioNano CMAP. These are those optical maps used for scaffolding that Olivier was talking about earlier. We're going to have 3 FASTA files, which are our hi-fi synthetic reads, or hi-fi reads, that's all you need to know. And then we're going to have a data set of Hi-C reads, a data set from a Hi-C experiment. And since those are Illumina paired reads, you might be familiar with the 1_1 for forward, _2 for reverse notation. So we're going to import these by, first of all, let's import the hi-fi one first. And then so we're going to click all the ones that say hi-fi at the beginning and then do export to history as a collection. And then leave this list checked off, that's fine. Scroll down to where it says select history and make sure you have the VGP workshop or whatever history you have selected. You should all just have one history at this point, so I think this is fine. And then click continue and it'll ask what you want to name this collection. And so we're just going to call it hi-fi data or whatever you want to call it. And create collection. So what that's doing is taking the shared data and just throwing it into your history so we can work with it. But we also need the CMAPs and the Hi-C reads. So we'll click those separately. Make sure you don't have the hi-fi ones checked anymore. And we're going to export those as a data set. And yep, this is good. Just import it to history. So the difference between how we imported the hi-fi and the other and the scaffolding data is the hi-fi is being imported as a collection. So these, the way Galaxy works, you can feed it collection data, such as this one, and it's going to run like a batch process. So say you want to run cut adapt, you can give it the whole collection, and instead of like telling cut adapt to run 3 times, it'll just know it's a collection and run it as a batch across all 3 of them. So that's what we use for the hi-fi data, since it's usually, we can have like up to 10, I've seen like 11 smart cells of data for something. Okay. So now we can navigate back to our history. Yeah, okay. Okay. We can navigate back to our histories. If you go to user, this is the view I like to use. If you click user histories, you'll see all of your histories. And if you click view on that one, then you'll see what's inside it. It's also present on the sidebar. Okay. So the next step is creating that K-mer database we were talking about earlier. So we use a tool called Merrill for K-mer counting. And yes, that's it. We're going to use a 31 K-mer database. So you'll see the...
Sorry, this K-mer counting process is really what I was doing before with the 2 sequences, except that you do it for all your reads, no? 1 by 1 for all your reads.
And instead of just 3 little instead of 3 bases, we're going to be using 31 bases. We're going to use 31 K-mer database for this one. So if you click in the tools panel, if you search for Merrill, click either one works. And click on the tool, then you can see all the various parameters that you can customize. So Galaxy provides this as a GUI that you can just edit more easily. And whereas if you wanted to run a Merrill in the command line, you'd have to feed it as flags and various parameters on a one-liner. But here we can just edit it as if we're just like running any like very simple to run tool. So we're gonna use count because we're counting the K-mers. And yep, this is all good. We'll just canonical K-mers, which are the ones that are not error K-mers. The cutoff is...
No, there's no cutoff.
Okay. So for your input sequences, we want the collection. We made a collection, so we need our hi-fi collection. So click this folder icon, and then your collections will be available now. So you only have one, so it should autofill with the hi-fi data. And then for k-mer size, we're setting a k-mer size, and just type in 31. So yes.
Good question.
Well, this was the one for the yeast. We usually use 21 for the vertebrate ones. It provides like a good, it's robust enough to cover most of the genome and most of the unique parts without being like super computationally intensive, because the longer it is, the more time it'll take.
So one reason to choose the k-mer length, it really depends on the sequencing technology, particularly the sequence quality. Because imagine that you have a lot of sequencing errors. So if you have let's say one, like remember the Q10, which one error every 10 bases, it means that no k-mers will really be real, no? Because if you use 31, because there is no single 31 segment that is actually not affected by a sequencing error. So instead, if your your quality score on average for your reads is Q30, which is 1 error every 1,000, then you find tons of k-mers that are correct, and every now and then you find some that are wrong. So if you remember though, the way the distribution looks like is a sort of balance between the first spike, not the very beginning, at around 0, this one, and the others. And depending again on the quality of the sequencing data, it will affect how much you can. So basically, it's been shown that 31, between 21 and 31 is good for this kind of analysis when you have either hi-fi or Illumina reads that have a comparable quality score.
All right, and the last thing I just want you to set is this job resource parameters thing. This is something that just as I said, this instance sends these jobs to our cluster. And so this just lets you change any of those settings if you want to use 32 cores, if you want to use 16, 2, etc. So we're just going to use 4. So click on this dropdown and click specify job resource parameters, partition VGL, that's good, and set cores to 4. And if hit execute, and then we'll just wait for this to run. It'll take like a few minutes, not too long. And in the meantime, I can pop open terminal and we can see maybe our jobs running and hopefully not crashing things. It's hard to test things when you have like 2 people using this thing, but let's see. There we are. See, everyone's here. We flooded the cluster, and that's fine. That's what it's there for. Look at all you guys running things.
So the cluster is here, no? I think it's the one next door.
Yeah, so that's why it's so fast. Sorry.
If you're not faster, it will go. Yeah. Okay. Let's see.
If you smell smoke from the air, you know why.
That's why we keep having fire alarms. The rest of these, okay. I don't know if you guys want to grab like a quick snack, a pre-snack snack.
Questions first?
Yeah. I mean, like it's done already, some of them. Yeah. Like mine finished.
What question? Or you have one question?
Okay. Like, see all of these cleared already? Hopefully that means they worked. Mine was green. Is everyone else's green? Oh, thank God. Let's see. Let me check the chat on Zoom. You gotta start in the cookies. Let's see, chat. Yeah, everyone else is green. Keep this open for a little bit. Let's see. When it finishes, if yours is already done, what you can do is we're gonna just rename it just so we know what stuff is what. So where it is.
So when you have a successful run, you're free to take some cookies.
Yeah, there's like snacks outside, but if not, just wave me down. But it'll want to rename it first. I'll just click this and then here, and we're gonna rename it as collection Merrill DB. And if not, it's fine. We know which one that is. Go for it.
Take a 20-minute break. Also,
Yeah,
for those
for
online,
Zoom.
20 minutes, no more than that. So we're going to resume at 3:40, okay?
Okay. Sorry, we couldn't give you guys cookies over Zoom, but that would be very logistically heavy.
Okay.
Or you could come twice and then go back to wherever you are if you're on campus.
Click on it. And then, yeah. Yeah, sorry, that was a little there. So the rest of the data from there, we named them. What do we call it? Collection.
So just to recap, what we just finished before was generating our Merrill database from our hi-fi reads. And before we left, I was just saying it might be helpful to rename the database. And so to do that, you're gonna have to click the name here. I've already renamed it, so yours will look different. But click it, and then it takes you into this little view of just a collection. And then when you click this pencil icon, then you can rename it to, in this case, Merrill collection Merrill database, and then save it. And so that's just kind of so we remember what everything is. It's titled with a job name in the output, but it's human readable names, makes it a little more friendly to look at when you're looking at your history. The questions in chat, we're good? Okay. So the next step, now that we have, so these are the counts from each, this is the count for each FASTA file. So in this case, probably each smart cell. We want to combine these so that we can have it across all of them. So what we want to do is we're gonna run Merrill again. So it might still be in your toolbox. If not, just search for it again and click on it. And we're gonna change this count operator to operations on sets of k-mers, because we already have our k-mers databases. We have 3 of them and we want to do something on them. So we're gonna do operations on sets of k-mers. And then for, we're gonna do input Merrill database. Yeah, sorry. We're gonna do a union sum is return k-mers that occur to any input. then set the count to the sum, because we want to get that union sum across all 3 of our individual databases into one. So union sum, and then for input Merrill database, we're going to click the collection again, and you're going to click your click your collection Merrill database. What are you smiling at? And then job resource parameters, just the same thing as before. Specify job resource parameters, VGL, 4 cores, and this should be good to go. Execute. And this one might take like a couple seconds as well. Let's see everyone running their jobs. I've got a few of them here. What is all of this? Oh, that's me. Oops. See, the thing about it, when it's a workflow, it'll queue stuff up. And so I didn't mean to take up some of the nodes today, but I have a couple that are going. And so when it finishes one job, it might issue 3 more jobs. So that's why that's taking up some of the nodes that I didn't mean to. Oh, well. So let me see. Anyone still? I think we're good to go. People are green. Good. Oh. This question in the chat. I can click this and just have this up so you see what you're supposed to run in the meantime. People whose it's finished, you can rename the output if you want. So this one, I think you can just click the pencil here. Yes, you can click the pencil and rename it. We're going to name this merged Merrill DB and save that, because this is the one that's merged from across your 3 different Merrill databases. I think I will move on in the meantime while Cassidy and Julio help out a couple others. There's some more questions in the chat, so I think we're good for now. So now we have our merged Merrill database, and we're gonna run Merrill. We're gonna run Merrill one last time to generate a histogram. That's the actual file that gets fed into Genome Scope. So click on Merrill again, and then for your operation type selector, you're going to want to do generate histogram data set. And then it's just going to be your merged database is your input. And you can keep this as this, or you can change it. We'll set it again to 4 cores. This is fine. 4 cores. So this is what you want to run. I'm going to execute it, and then like, so this is what you have to do. Keep open our chat. And for people who have finished, you can go ahead, and if you want to, you can rename it. This is going to be your Merrill DB histogram. We're just gonna, so again, these are the parameters you need to run to generate the histogram for anyone who's still doing that.
We'll take a second, exactly which step is the.
Sure. So I'll just scroll through on this side. So the first step was running Merrill DB, the k-mer counting to get a database of the k-mers in each FASTA in this case. So this is the one for the first one, second, third. But then we don't want them split across them. We want to merge them so that we can actually like look at them all together as a unit. So that's the step that merges them together, and you have your merged Merrill database. And so this file format is like, you'll see it says compressed binary. It's ugly. We can't put that directly into Genome Scope. So the last step is just a conversion to get the actual histogram, which looks like more visible, I believe. Let me see. But this is what goes into Genome Scope. Let me see if I can view it. Because this one is just a plain text file. Let's see if you work. Yeah, you'll see that this is the histogram file. So these are k-mers at coverage 1, which are probably the error ones. And you'll see it goes up to the middle, a little peak, so forth. So this is what Genome Scope will use to plot. Because the database file itself stores like information about the k-mers, which isn't necessary. We just want the counts. Okay, I think I'll move on to the next step. That's okay. Cool. Let's see the next step. Now we have that histogram file I mentioned we need for Genome Scope. So the next step is running Genome Scope. So we'll search back in the tools for Genome Scope and just click on it when it comes up. So our input file is that MerylDB histogram from before. Ploidy is going to be 2. And the k-mer length, it defaults to 21. We'll change that to 31 because we use 31 to generate these. And that should be up. And last options we need, if you'll scroll down, you'll see in the output options, you're going to click on summary of the analysis. This just gives us the summary, the characteristic summary at the beginning. It gives it in a text file so we can actually parse it later on. And for advanced options, we'll click here to expand this, and we will click on create testing TSV file with model parameters. This again is just creating another file that we can parse later on for some values that we'll use for downstream. So I'll leave this up. This is what should be inside this input. Okay, and then sorry.
Can you go back to that? Sorry.
Yeah. This slide on the side is just showing what you should be changing in here. If something's not mentioned here, then leave it at the default. So what would be changing? Let's just say 31. What should be changing is that to 31. We're gonna give it our input file. And then we just want to change. We're just having it give a couple more outputs. We want this summary of the analysis within output options. And when you scroll down to advanced options, we also want a testing TSV file. And then that should be good to go, and hit execute. Let me see if there's anything in the chat. Good so far. Everyone's Genome Scopes are running. And it'll be like very quick because mine just finished. So let's just take a look at what it looks like so far. Let's hide this. And so what the plot we've been showing you guys so far has been the linear plot. There's also a log one, but this one, the log one is useful for when you want to see further at super high coverage. For instance, bacterial contamination, like Julia was mentioning earlier. But for getting the gist of the genome, this is fine, the linear one. So this is what you should be getting when it's finished. We have our summary up here. So it's 11.7 meg, 93% uniqueness, 0.58% homozygosity, this little peak here. You've got 25 at haploid coverage. So that's going to be 50x diploid coverage. And this is a nice little bimodal distribution for this diploid yeast. And this is our much smaller error profile, which I'm presuming is much smaller because these were synthetically made. So I think Krista just made them look nice, which is fine. This will usually be bigger if in real data. So this is what our genome scope looks like for our test yeast data set. So as I said before, we're gonna need to parse some of these parameters because, for instance, we want the genome size for later on. We want to use that to help calculate NG50 and the NG statistics. So I'm going to pull up my slides for those steps. So we'll use the estimate. Yeah. So basically we just do some text parsing, text manipulation. This is where if this was a bash script, it'd be stuff like cut, set, awk. But Galaxy has those packaged up as these tools as well. So we're going to use, for instance, replace parts of text, which you might be used to as grep. Replace parts of text. And it'll be this one. Click that. Let me make this smaller. Okay. And then so you want to give it the file to process. It's going to be your genome scope summary. So this is one of the summaries that we asked for, just so that we have everything in a text file that's actually operate-onable. For the find pattern, we want to do just BP. It's going to look for that string that says genome size BP. For replace with, want to leave this empty. Replace all occurrences of the pattern. Click on yes, and click find it. This is fine. Find and replace text in an entire line. That's good. So go ahead and hit execute when it looks like this. There. So let's just, if you want, this is the summary. This is the file we're working on. So it's looking for BP. So it'd be like haploid length, looking for BP here. That's all this is doing. And then next, it's just a couple of these sorts of steps. So bear with me for a minute. Next one is replace parts of text again. And our input this time is our replace from the previous job. So it's just this. It should be filled automatically because that's the most recent text file. For the find pattern, you're going to type in a comma and leave replace with empty and click yes again on replace all occurrences. And then this should be good to go.
And in here is just because the output is formatted with the comma.
Yeah. So the workflows that we have will do all these automatically, but we have to do this like this for now. Search. I do not do this every time. I have to let you know. Search in text files. We'll have grep. And we're gonna select lines from your previous output. It should be auto-filled. That's that replace. Search for ones that match. For type of regex, we'll switch this from Perl to basic. And regular expression, haploid. So this is where we're really just getting that line where it says the haploid genome length. And go ahead and, oops, what's happening? Go ahead and execute that. Chat. Oops, okay. I will pause. It will slow down from now on. Let's see. Do you need, do the people in Zoom need me to go back to a previous step for now, or? Okay, previous one, that is like the one where we're replacing, or the first replace? Okay. So there's like 3 replaces happening. The first one, you're looking for BP. The second one is the comma. And then the last one's going to be searching for haploid.
Yeah.
Sure. The first, yeah, I'll just go over it again from the beginning. The first one, the first replace is base pair. Yes.
Next one is
So this is the first replacing BP. And then the second and third one are replacing comma, just comma. And then the last one is looking for, is not a replace, it's a grep, and you're looking for haploid. These are the parameters, instructions. Sorry? So this one, so grep doesn't replace, grep selects. So it might be more helpful if you look at the outputs throughout it. So this is our initial file. This is what we're feeding into everything. And then we want to replace, we took out BP because we just want the number. Our second replace, we took out the commas because the stuff downstream can't process the commas. And then grep is just getting our genome haploid length. And it's just, because that's all we need from the summary. The third one's using grep. It's replace, grep. Yes, deleting it, yeah. Yeah, sorry, it's a little unintuitive because like the Unix tool that would be sed, which can delete, or it's usually used for replacement. It's replacing it with like nothing to delete it, yeah.
Why do you care about removing commas from the number?
Because the stuff, when we put it downstream, when we give it, we're gonna feed it just this number directly, it doesn't want commas. Like it'll just crash.
Can you show the?
Sure. So to grep, all you're changing is the type of regex, and we're searching for the term haploid because we just want to pull this line that has the genome haploid length. Okay, I think I'll move on to the next one. And if anyone on chat is having problems, I can also help you during, or Natalina can help you during the more theoretical lecture after this. So the next step after that, so this is what we have right now, this file that has genome haploid length, tab number. And then also, if you're wondering why there's two different numbers, it's because the summary estimates a min or a max of that property because it's a model. And we usually, I think, just go with the first one. So the next steps, so this is what we're working with right now.
They're very close to each other.
Yeah, in practice. Similar. And since it's only estimated anyway, let's see. Okay, so to actually get this number isolated, we're gonna use a tool called convert delimiters to tab. So search convert delimiters to tab. And all that we're changing here is just, well, actually, we're not changing anything. It should be autofilling with our most recent file, which is that search and text files output. And this is just converting to white space because it may be, for instance, spaces when you want tabs, because that's more easy to parse. So that's just all this step is doing. It's just more text parsing. So this should be filled fine automatically. Execute. And this output will look similar because all it's doing is changing the white space. But we'll take a look anyway. Well, it changed our white space on here. So those spaces in genome haploid length became tabs. And now that we see everything is in its own tab, like in its own column, we can just tell a tool to pick column 4, which is what we're gonna do, or 5, which is what we're gonna do next. So advanced cut is the tool we're gonna use to do that. It should be here. For the file to cut, it's gonna be this convert output that should be there automatically. We wanted to keep it delimited by tab, cut by fields. And in the bottom, you'll have this list of fields option. If you click in here, it'll give you the options and just click column 5. We want that. We want the number that's currently in column 5 of that file. And then we can hit execute. And when we check up on that file, it should be, yep, we just have this little number, which is our estimated genome size according to genome scope. This, everyone should have this at this point.
Which is what I expect for at least 12 megabytes.
okay? Zoom people will be okay? I need to use my coffee image. No, I have too much of this. The Purge Dukes parameters. Okay, I think I'll go on now to the next one. So this is just the genome size. A couple other parameters we use are related to coverage, because we'll talk more in depth about it when we get to purging, but that's going to be, so when we run contigging, sometimes you might not get a perfect primary assembly. Sometimes it'll have parts of, sometimes it'll have what we'll call haplotigs, which should really be an alternate instead of being represented twice in one assembly, because they represent the same heterozygous region of the genome, but perhaps they're so different that the assembler thinks they're two different parts of the genome and puts them like there together in the same assembly. So we want to remove that so that we have a proper, complete, not duplicated, full primary assembly. And that's the purging step. And that relies a lot on coverage, because we're going to be mapping our reads back to the assembly. Because if, for instance, you'll have duplicated regions, I see a dip in coverage, or like repeats will have, the cutoff will have a very high coverage cutoff. So we get those coverage cutoffs and the transition between haploid and diploid. We're going to get those from our genome scope parameters as well. Oh, can we, okay, I'll share this. Let me share the slides real quick in the Zoom. I can just share this. Yeah, okay. Let's see.
You could also share, or I can share the slide.
Yeah, that works. You have a, can you do that? Okay, thank you. Okay, so Cassidy is sharing the slides so we can follow along more easily. Sorry about that.
But I can't do it in Zoom.
Put it in the Slack. I think that's fine. She's gonna share them in the Slack and I will share them on the Zoom. Share, or maybe I don't have permission to. Okay, let's see. It'll be in the Slack with, oh, that's a lot of people. Thanks for joining. Okay. So I think everyone's okay so far. We'll do the purge parameters. So this is just more text parsing. I'm sorry, this is the boring part. So the first thing, okay. What did you do? What did you do?
We ask image information so that people can view it.
I see. Okay. That's fine. All right. So to get our purge parameters, the first step is getting the maximum read depth. So this is going to be our cutoff basically for like anything past this is like going to be filtered out as a repeat, as like a possibly a collapsed repeat, because it's getting way too many reads piling up on that region of the genome. So to do this, the first step is going to be running compute an expression on every row. So compute. It's going to be this one up top. Keep this open. So this expression is what is actually the math it's going to do on the given columns. I'm not sure why it's rows then, but we're going to type in 1.5 times C3. And we're going to do this as a new column to genome scope model parameters. So we're going to have to click in the drop down and make sure you click the model parameters. Round result, click yes, and leave this as no. So I'm going to hit execute just so that this runs. And so what this is doing, let's take a look. This is the model parameters file. I don't remember the format of this file. Shirley, do you remember what the columns mean? Okay. Well, this one's the haploid one. This is what I think. This is 25. That's our haploid coverage. I just don't remember what the other ones are. One of them is error. Yeah. Well, we're operating on our haploid one here. So we're getting basically 1.5 times it, and we're gonna count that.
So what's the ratio? 1.5 times the haploid average.
This is from the Canoe paper.
So what's
Do you remember?
So essentially you want to distinguish everything that is sort of haploid or diploid from the rest. And if you imagine that the well, it wasn't,
Let me
it
pull
wasn't
up,
experimentally determined, but if you imagine more or less that 1.5 is that peak over
the dip between the two peaks.
and if you do 3 times that, it's actually
It's going to be over here. So we're just trying to get like this transition parameter and our maximum one, because everything above maximum will be in per groups counted as a repeat. Got a couple chat questions.
Yeah, so it was already.
Yeah, it was empirically determined. But the idea is to get some value that says, okay, anything above this is repetitive. And with repetitive, even just a few copies more than 2 copy versions.
And then the 1.5 times, that's like.
That's going to be the transition parameter. It's going to be here, this little dip. So like sometimes, so it's also used with the coverage because like when it's per.
1.5 is actually a transition.
Yes,
So-
that's what I mean. Yeah.
So 1.51 would be haploid, 1.5 is between haploid and diploid. Diploid would be 2.
Like when it's mapping the reads back to the genome, like it'll be able, that's like where it's going to cut off between a haploid and diploid when it observes the coverage in there. That will, it might make more sense when you get the actual purging. Sorry, there's another histogram later on. Where were we? Okay, so that was getting our parameter. Let's see. And then the next one, we're going to do compute an expression on every row again. This one's going to get our repeat parameter because it's times 3. So we're going to, the expression is going to be 3 times C7. And adding this as a new column to this compute, like the previous step. So it should be auto-filled. So just type in 3 times C7 and round result, yes. And then that's good to go. To execute.
And so 3 times now you're doing that with the diploid value.
That was the transition one. So it's really like 3.5 times. Sorry. That's the transition parameter. So it'd be like,
Okay, so this is the one I was actually referring to before that, you know, it's
be
a
here,
large
1,
value that
2,
it
3.
will start to include all the repeats.
Anything like past, like here, like when we see it, it's gonna be like 118. So it's gonna count over stuff with coverage over 119 as like a collapse repeat. So that's what it means like in practice. All right. So we have our.
Sorry, this is not super clear now because we don't have an assembly yet. We're only parsing the raw data.
Yeah, it's very abstract now, sorry.
But then we use this to work on the assembly.
Help us identify what is repeated, what is not repeated.
It's an expectation.
The 1.5 is 3 are the constants that you use for all the information.
Yeah.
Yes.
It's not like.
It's just, you know, the basic assumption, if 1 is the haploid, 2 is diploid, and the peak.
And this,
so
yeah.
usually-
And then the 3 is just, it'll capture us, but yeah. Okay, we can rename this just to, because we have a couple compute now, so we can rename it so we remember which one we're working with. We're renaming the one that we just finished. And we're gonna call it parsing temporal output. And then we're gonna use advanced cut. And we're gonna cut this parsing temporal output file, the one we just renamed. We're gonna cut by fields and we're gonna get column 8, which is the maximum def that we just had. So we'll execute this, and when it's finished, then you can rename it maximum def so that we know when we're looking at our history later and we need to use this later on, we know where to find it. So once it finishes, then you can rename it maximum def. And then the next step is also just running advanced cut again. So just doing the same thing again on parsing temporal output. And now we're gonna get our transition parameter, which if you remember was column 7. So it's just this, execute, and we can rename this.
This one's column 7.
Yes,
What was the first column?
the first one is gonna be column 8, and you can rename that as maximum def. So column 7 is gonna be transition. Yeah, and so this is some of the output we'll see later on. So this is one of the histograms that produces, and it's a little hard to see. Maybe I'm only a viewer, so I can't make it bigger now. But well, these vertical lines, this is the coverage. This is the coverage histogram generated when you map all of your reads against the assembly you're trying to purge. And these vertical lines are going to be these parameters these cutoff parameters that we just found. So this one here is going to be your transition between haploid and diploid. Here's all the haploid, here's all the diploid. And then this one's going to be that maximum def parameter. So anything, if you see like a spike here, we'd be counting that as a collapse repeat, and it'll be removed. Yes, Rita.
One question.
Mm-hmm.
When we did the last step with transit that we ended up renaming transition parameter, we were operating on parsing temporal output,
Yes.
but that was the ×3. I thought the transition point was the previous one.
So parsing temporal output, it should have the times 1.5 and ×3 in it. It should be column 7 and column 8. So like when you finish, it should have, if you look at the history on this side, if you click on them, you should have maximum def is 114, and then transition parameter is going to be 34, I think. Just wait. Yeah, 38. And then this is just our setup, our extremely boring, I'm sorry, setup for tomorrow when we do purging and a couple other things. And there's more plots that we can look at. Okay. This was when we were supposed to have a break, but we took it earlier.
No, right.
We don't have time for that.
Thank you, Cassidy. Okay. Let me, yeah. Slideshow.
Okay.
How do I present this now?
So now, before we start assembling the genome, I'd like to give you an overview of how, in practice, in terms of algorithms, we assemble genomes. Good. Thanks. All right. Remember this slide from my first presentation? We had these two approaches to completion of the human genome: hierarchical shotgun sequencing, which we don't use anymore because this has this BAC and YAC intermediates, and whole genome shotgun sequencing, which is really what we do. But of course, that's more challenging from an algorithmic perspective because there are way more overlaps that you have to compute. Again, the idea, we have to find overlaps. That's the process of genome assembly. Imagine we have all the sequences, we start to arrange them, trying to find, like pieces of a puzzle. Essentially, we need to find the suffix of a read that matches with the prefix of another read. The suffix of, for instance, this read that matches with the prefix of this other read on top. And as long as we are able to do that, now we can arrange them and determine a consensus sequence, which is the original sequence. And again, the reads overlaps because we're sequencing, multiple times the same regions of the genome, and we can find these partial overlaps. So there are essentially two main approaches to genome assembly, and then of course a variety of approaches that use one of them in slightly different ways. So the first one is this OLC, overlap, layout, and consensus, or like string graph-based assembly, which constructs overlap graph directly from the reads, eliminates redundant reads, because of course you want to try to make it also fast process. We have computed really fast, but still we're talking about hundreds of thousands or millions of reads. So we have to do something that can finish in a lifetime for sure, but also in well, that's but also in a few hours if possible. And then you create, and we're going to look in detail in this, but then trace a path and essentially reconstruct the original assembly. The other approach is this DBG, the Bruin graph based assembly. And as you can see, it uses skimmers induced from the reads, basically discarding the original reads, okay? And again, we'll create some kind of graph. So what's behind graph theory? There is this famous example from the mathematician Euler, which is not the famous one, but this is from the 18th century. And apparently it was looking for this problem of the bridges of Königsberg. And essentially, here you have the city where you have multiple bridges and you have different, of course, areas of the city. And the question was essentially, if you want to walk around the city, is it possible to visit every part of the city, walking through these bridges, but essentially only return exactly once on each bridge and returning to the starting location? So that was the idea. And also, if you simplify this, and that's why we start using graphs. Now you have these 4 areas of the city. You have the bridges that connect them, and the question is, can you really go and touch all the different areas of the city without going twice in the same place? So that was the sort of intuition, and that's also the way this OLC approach essentially work. So again, Roger Staten, I presented him before, 1980s, was the first sort of to describe this OLC approach. So we're first, of course, looking for the overlaps between the reads. Okay, so we have all this bag of reads and we need to first identify the overlaps one way or another. Now I presented the alignment approaches before, some that are using like k-mers just to infer what the most probable, what is the most probable overlap between some of the reads, and then you can compute exact overlaps after that. Then you create a graph that really connects all the reads through these overlaps that have been identified. And then you have to find something that is called the Hamiltonian path, which is really what also Euler was looking for. Now it's a path through the graph of these overlaps that goes and touches essentially each read only once. Okay, so each node in the graph only once. And if you find this Hamiltonian path, then you can essentially reconstruct a consensus sequence that should represent the overall assembly. So, okay, so basically, usually you do an all versus all attempt. Now, essentially, you're trying to, because that's the de novo, the power of this whole genome shotgun sequencing approach. You have all these reads and you know nothing about them. You have no space relationship between them. So what you do, you try to compare all the reads versus all the reads. Again, you take shortcuts like using k-mers to try to identify the most plausible set of reads that overlap between each other. But in the end, you have to compare all of them somehow. And then once you have all these alignments, you merge them together in this overlap graph, which is a directed graph. Now, directed means that there is a sort of a direction between one read and the other read. And in this graph, again, the reads, this is an important distinction between the OLC and the Bruin graph approach. The reads are the nodes, and the links or arcs or edges, they have multiple names, are the overlaps. So they tell you, this node, which is a read, connects to this other node, which is another read, through an overlap, which is this arc, edge, or link. Okay, so you always have to find a cutoff to define overlaps now, because imagine if you're saying everything that has even one base in common is an overlap, then almost every read will have tons of overlap. So you need to find a minimum threshold to define something that is a real overlap. It could be 5,000 bases if you have long reads. It could be much shorter if you have short reads, and so forth. So for instance, in this example here, the minimum overlap is 5. So if we define as minimum overlap 5, and imagine we had this original sequence, we have generated 6 reads out of it, then we can construct this graph that represent the relationships between these reads once we have computed all the possible overlaps. So of course, you can see that because of this threshold, for instance, some of the overlaps, even if they actually exist, now you can see that even read 1 still overlaps with at least read 5. Yeah, so you have a TAT terminal sequence on read 1 that overlaps with read 5, but we do not represent these overlap in this graph because we have set a threshold to 5, right? So this is an OLC graph, okay? And then basically, once you have defined this, essentially you have to well, usually represent also the length of the overlaps in these edges. Now you basically tell the software how long is the overlap for each of the edges that you have. Sorry, this is maybe we can look at this. This is a real sort of real example. I imagine you have a short sequence like this, and you generate these 5 reads, okay? And then you want to you look for the overlaps between them, so you can see the red arrows, they correspond to the overlaps. And what you really need to find is a path through these overlaps that leads to the original genome. That's the next step. So yeah, while we reconstruct the original sequence, we have to find a path through this graph once we have the graph, okay? And these, of course, there are multiple paths that are possible. And so we have to really find, you know, this, essentially this path that starts in a particular node and ends to in another node, visiting each node exactly once. That's the definition of a Hamiltonian path. Imagine this example now where we have this very simple sequence. Okay, that's the original sequence. We define overlaps as those that have at least 4 nucleotides that overlap. And imagine we have generated these reads. Okay, so if you essentially compute all the overlaps, then you can draw this graph, okay? So for instance, if you take this GTA-CT, there is an overlap of 5 now, because if you look at this sequence here, TAC-GT is the same as here, TAC-GT. The last 5 nucleotides of this sequence are the first 5 nucleotides of this sequence, okay? So then, you have this graph, and for instance, in a very simplistic approach like this one, we could just say, well, let's start from this node, okay, then walk to the other node. So it means essentially the first nucleotide is going to be G. Then we have TACGT. And then we have a perfect overlap of 5 bases, but then there is an extra base in this other read, which is A. So let's add this one. This has another overlap of 5 with this other sequence. And so we basically add an extra C, then we add an extra G, an extra A, and finally a T. This is the Hamiltonian path. The question is how you find it. Turns out that Hamiltonian paths are sort of what's called an NPR problem in informatics. So there is usually not like an optimal algorithm that finds a perfect solution in a linear time or an efficient way. So basically, then when that happens, you need to find algorithms that find optimal or suboptimal solutions in a reasonable amount of time. So one approach is this shortest common superstring. It's not really ideal, but it gives you usually a good approximation. So basically, we're given a set of strings, like in this case, okay? Basically, we need to find a shorter string that contains each of our input strings as substrings. So imagine you have this, these, let's say, reads here, CTG, GAC, ACT, and so forth. The shorter common superstring is what you can see here. So imagine that you start from the left. So CTG is the first sequence, but there is an overlap between this G and this other G. So basically we collapse them. So we have only one G, so C, T, G, and then CA, then there is also another overlap here, which is another A. So basically we skip that A, and then we add CT. And then again, T is overlapping here with T and so forth. So this will be the shortest common superstring. The problem is that here orders matter. So if I change the order of this 3 MERS, essentially I get a different shortest common superstring. So I need to find the shortest common superstring that is really the one, the real one from the data. And so that's how you achieve that in practice. For instance, there is this greedy shortest common superstring approach, which the word greedy usually means that it's not finding the optimal best possible solution, but is a fast way to get to a potential optimal solution. Again, you don't have a certainty about that. And essentially, the way this works is depicted here. So essentially, you start by combining the strings one by one, starting from the ones that have the best overlaps. So imagine you have this graph here with AAB, ABB, and so forth. All these are the overlaps. So these are the 2 as essentially the longest overlap. And again, here there is a decision you have to take. We'll start from this particular overlap between AAA and AAB, and you can see you can combine them into another sequence that, because of their overlap of 2, is going to be AAAB. Okay, so the first A here, the 2 common A's, and another B. And now we have simplified the graph, right? So we can now pick another edge or another overlap that is long. Okay, so in this case, for instance, this one, and we combine them as well. So we get this new sequence ABBB from ABB and BBBB. And then we combined ABBB with BBA. You see that they are directional. So this overlap is different from this other overlap. This has only one base, the last A here, but not the other one. So we combine them, and then we find this shortest common superstring using this greedy approach that again combines the strings based on the longest overlaps sequentially. Again, this is not going to lead to the best possible solution. There are better algorithms to find a Hamiltonian path through a graph, but this has been actually used in some assembly software. So, of course, repeats are, like we said, a problem. Here is an example that exemplifies essentially why this is a problem if you use greedy algorithms like this one. So this is a popular example when you have a string that is repetitive, so a long time. This is actually a repetitive string. So if you imagine you take all the substrings of length 6 from this particular sequence, now these are sort of sequencing reads if you want, you can see them here. Basically, if you apply this greedy algorithm, what you got back from this, given a length of 3 as overlap, is a long time. So we're missing a single unit of the repeat again, because essentially our length of the overlaps is too short. And this is what really happens in genome assembly, and this is why having long reads is really superior to short reads. Because if imagine this situation, we have 4 regions of the genome, L1, L2, L3, and 4, and the blue portions here are unique sequence, so something that is not really identical. They can be easily distinguished. And then you have the red part here that is actually the repeat, so the same sequence in all 4 regions. Okay, so then you start to create these graphs. And what happens when you hit the regions that is repetitive is that essentially all the graph have overlaps that converge into the same place. And essentially you don't know how to walk through the graph in these regions because there are too many overlaps that are potentially valid. Okay, whereas if you have long reads, then you can essentially span them, and they essentially are not repetitive anymore. Going back to the OLC, essentially, so we have now covered the essentially the overlap approach, the overlap step. So we now have this layout step. There are a few more things that you can do once you create the graph, and they are usually taken care by the software that essentially simplify the graph. Even before you try to find this Hamiltonian path through the graph, you want to try to simplify it so that it's easier for the algorithm that needs to find the path to find a reliable one. So these are little tricks. So for instance, the first one is to remove what's called transitive edges or redundant edges that essentially skip nodes in the graph. So imagine you have this situation, or you have 3 nodes, again, 3 reads, and there overlaps, and there is an edge that connects essentially the first one to the third one directly, or the first one to the fourth one directly. That's redundant information in the graph because you already have the other edges that tell you exactly the same thing. But if you keep this one, then you cannot go back, right? Because remember, the idea of the Hamiltonian path is that you have to be able to touch all the nodes in the graph. So if you walk this way, then it's hard to go back and you may miss some of the nodes. So you remove these transitive edges. You prune what's called spurious branches that are usually sequencing error. So imagine you have one read that is different from the other for a particular region, again, because there is a sequencing error in it. This will lead essentially to an edge and a node that essentially has little support. It only comes from that read. And there is no need to really go to touch that node. And again, because if we want to try to touch that node, we have no way to go back. So again, we prune it and essentially get rid of most of these artifacts that are left in the graph from sequencing errors. And then we are left with non-branching stretches that correspond to our context sequences, okay? So that's the general idea. And I mean, there are a lot of tricks like this that essentially allow us to polish the graph. So here is the final step of this OLC, which is the consensus step. Here you can see, then of course, we still have some errors. So for each contig, we need to essentially select the most likely sequence because again, we still have this overlap graph, but it's not like a final sequence. So what we do is we pile up all the reads that make up a particular contig and verify that base call is supported by the majority of the reads. So for instance, here we have a deletion that is only present in one of the reads within this overlap graph. So we decide only to keep the majority and essentially disregard the deletion, and also of course all these point errors here and there also get removed in the final consensus. So in terms of limitation, this is very demanding from a computational point of view. It's very intensive because you have to compute all these overlaps and can be therefore very slow, particularly if you have Illumina short reads, because you have millions of reads and you have to compute all these millions of overlaps, of potential overlaps. So how are we doing with timing? Good, okay. So, the second approach that I mentioned before is this the Bruin graph, which is a different approach. It's counterintuitive. It was essentially introduced a while ago in 1995 by some other people, some people that were already working on this problem of alignment, but was not really useful or used until the advent of Illumina short read sequencing, when again, because you have so many reads and they're all very short, there is a problem in terms of computing overlaps. You want to speed it up, and it turns out that the Bruin graph approach is very, very efficient in that sense. So the idea is to disregard the reads entirely. So you chop all the reads into their constituent k-mers, and then use all the k-mers to generate the graph. And again, remember, the Illumina reads are very short. So even if you chop them into k-mers, you're not losing so much information. It's also, again, a directed graph. And here, again, important distinction is that the nodes, they represent k-mers imagine you have decided that the length of your graph for the Bruin approach is 31. So the nodes will represent k-mers of length 30. So the length of the that you've decided minus 1. And the edges will represent the overlaps between them. So essentially, the edge really represent the k-mers of 31, and the two nodes between the edge represent 31 minus 1, so k minus 1. So let's try to look at this. The reason why this is actually more efficient than the Hamiltonian approach is because the Hamiltonian approach, again, I defined it before, it's an NPR problem, whereas finding a walk through this kind of graph, it turns out from a mathematical perspective is much easier. It's called slightly differently, it's called Eulerian walk. And essentially you have to be able to walk through the graph. But the importance here is not on the nodes, but it's on the edges. So you don't have to touch all the nodes at least once, exactly once, like in the OLC approach, but you have to touch all the edges exactly once. So in this case, again, from a mathematical point of view, being able to walk a graph, making sure that you touch all the edges exactly once is much easier than walking all the nodes exactly once. That's why the De Bruijn graph actually works better than OLC, especially for complex graphs, such as the ones that are generated by short-read sequencing. So, yeah, so here is an example, a very basic example. Again, remember now the edges are the k-mers and the nodes are the k minus 1 k-mers. Okay, so here we have AA, AB. Okay, these are two k minus 1, and the edge between them, that connects them, is going to be the 3-mer between them. So, so in this case would be AA, so AAB, and here you can see there is another edge that is connecting to itself. Okay, so it's connecting AA to itself, and again here we have to find this Eulerian walk that goes through all these edges exactly once. So how do we do that? Again, there are algorithms to do that, but in this simple example, we can figure it out ourselves. So we have to first go once through this edge. So it means goes from AA to AA. So there is a k-mer that is AAA between them. So that's how we generate this first 3 bases here. Then we have to go from AA to AB. So we basically add a B to it. Then we have to go through this BB. So we add another B to it. Then there are 2 edges again on the same node. And so we add another 2 Bs. And then we have to have a final A, and that's the final A in the graph. So it's a different approach to use graphs to the same goal. Yeah, so that's basically the same thing. This leads to essentially different results. So if you compare a graph from OLC to a graph using the De Bruijn graph, to an assembly that uses the De Bruijn graph. For instance, here is an example. If you look at repetitive elements, how they are represented in the OLC versus the De Bruijn graph. Okay, so here again, you imagine you have this region of the genome, okay, which in green and blue, it's the unique portion of it. And there is another region of the genome that has, again, a unique portion in yellow and in orange, but then there is a red region that is common to both. And it's the repetitive element. So if you build, an idealized graph, an OLC graph from this region, this is how it's going to look like in the, again, OLC space, whereas in the De Bruijn graph space, it's going to look like this. And again, because these are the original reads and their overlaps, whereas in the De Bruijn graph, they are chopped into k-mers. So they look different. And, this leads to a lot of different, different behaviors of these graphs, and they have to be treated separately. So going back to the Bruin graphs approach, essentially it's more simplistic in a way because you don't have to define a particular overlap length, which is going to be a very important factor in determining the topology of a OLC graph. In the case of the Bruin, you only really need to decide the camera length. When you have decided that, you will everything else is done, is determined. So of course that means that K is the most important factor in the Bruin graph construction and leads to different graphs depending on the length. So you can see here that if you lower K, you have more connections, because there is less unique sequence. But that also means that you have less chances to resolve small repeats. However, you also have higher k-mer coverage because, again, the shorter their k-mers, the more of these k-mers you find in their sequencing reads. On the other hand, if you have higher k, if you increase the k-mer length, then basically you have less connections. Your connectivity of the graph reduces, so it simplifies the graph. So you have more chances of resolving repeats. But you'll also lower your camera coverage, and it means that you can also miss some of the connections of the graph. And there is really no optimal balance here. It depends on the sequencing technology, on the complexity of the genome, and so forth. So yeah, to recap, the Bruin graph they have in terms of pros, they essentially transform the assembly problem in something that is tractable from an algorithmic perspective. So, with millions of Illumina reads, you can assemble a genome on a laptop in a reasonable amount of time. So there is no CPU-intensive computation of read aligning steps, because there is nothing to align. You just need to build this graph out of the k-mers, and there is really nothing to align. So there is no matrix of substitutions that needs to be computed, nothing like that. And you see the nodes and links number is approximately equal to the genome size. So it means that it actually scales linearly with the genome size. Whereas if you have an OLC graph, the bigger the genome, the more reads you need to generate, and every read essentially corresponds to a quadratic increase in terms of a number of lines that you have to potentially compute. What else? Yeah, there is essentially there is no consensus needed. So remember, now there was this step where you had to pile up the reads and find the optimal sequence. When you have dissected the genome into k-mers, there is nothing else left to be done. You just need to, if you find the Hamiltonian path, the Eulerian path through the graph, that's already your final sequence, your final consensus sequence. So it's the best solution to assemble large genomes with short reads, essentially. The problem is, again, if you have large repeats, the Bruin graph approach will fail into this and will leave your graph disconnected. So going back to the original problem of Euler, essentially the answer is no. It turns out that basically there is a demonstration that says that there is you need to have even number of edges through the nodes to be able to walk this kind of graph as Euler was trying to do. So essentially here you have, so if you take this node, or let's say take this node, it has 3 edges. This other also has 3 edges. This is what, 4 or 5 edges. So essentially, these are all odd numbers, and there is a mathematical demonstration that says, okay, there is no way you can walk this graph, by going through all the nodes once and also all the bridges. So that's not possible. Okay, before we start, just a few notes. There is a ton of assemblers that use these approaches. So here I have. I listed a few that works, for instance, for short reads, long reads. They mostly sort of follow this principle, and they're just different implementations. So these are all the Bruin graph-based assemblies. Some are even greedy. The string graphs one essentially are OLC. And then, of course, there are like slight variation on this same bead. And for long read assemblers, the jury is still out. There's, some people using the Bruin graphs, some people using OLC graphs. And again, it's still debated. What we're going to use, it's this software called IFASM that I was going to talk to you about a bit more. It's a string graph based, again, this sort of OLC based approach. The general principle is you have these hi-fi reads that will be described by Olivier a little bit more in detail, but essentially they are long, accurate reads. You correct the reads by looking at the pile up of the reads in the very first step, so not at the end, in the very first step. You identify sequencing errors, like in this case, this is only seen in this particular read, not in the other reads. So we correct this read to look the same. Here, instead, we find that there is consistency between a set of reads that all show an A, and there is another set of reads that all show a C. So we identify this as a heterozygous site, heterozygous allele, and we do not correct this, we keep them. And then we use these corrected reads that essentially show no sequencing errors to generate a phased string graph, which is, again, more or less a OLC graph, because these are overlaps between the reads. The reason why this is particularly effective is because, again, using the long reads and these heterozygous sites, we can phase both haplotypes. So whenever there is a divergence between the two haplotypes, we can keep track of it. So you can see that the graph here splits and essentially represents this region where there is this heterozygous site as a one portion of the graph, and the other portion of the graph represents the other alternative allele. Then you can walk the graph to reconstruct both haplotypes. So basically walking on this side of the graph, you get the, let's say, paternal allele, and walking on the other side of the graph, you get the maternal allele. Yep.
So, quick question about that. Do you have some sense for how, what density of heterozygous sites there has to be for that to fail entirely?
Yes. So, I mean, of course, that depends on different factors, but the general idea is that come on. So the general idea is that essentially you need to be, it's not going to fail. It's not going to fail. But the problem is that if the SNPs that you find are at the distance on average that is less, that is less than the read length, that's enough, to connect them and essentially create a chain of SNPs that allows you to walk entirely the genome side to side for each chromosome, okay? And then that's enough to phase the entire chromosome. Whereas if you have, even some regions of the genome where there is no SNP between the length of a read, okay, on average, then there is no connectivity. So you don't know, oh, should I now connect to this other SNP that is 100 kb far apart or not? So that's where you lose the connectivity between the two haplotypes. So if you think about the hi-fi reads that we're using in this case, they are about 20,000 base pair long. So if your SNP distribution, it's an average less than 12,000 base pair, then you start to have a problem because there will be less connectivity. So in fact, what is interesting is that nowadays, because we're using long reads, we prefer to have highly heterozygous individuals for this kind of reconstruction because they clearly separate out in the two haplotypes. Whereas for a long time, people were actually choosing highly inbred organisms because they had no haplotypes to reconstruct to begin with, essentially. Okay, yeah, that's it. High fidelity long.
Stopping here.
Yeah.
Yeah, it's like
What time is it?
5.
Okay.
So I think we can pick up again tomorrow.
Yeah.
With items. We'll start with the practical portion, which is just we're going to set high.
Okay, I'll repeat that. Yeah, go ahead.
We'll set HiFASM to run while Olivier gives his lecture because the HiFASM takes about 10 minutes to run. So we'll start off with that tomorrow.
Yes. So just to repeat this for folks online. So we're Instead of starting a new session that we'll probably not be able to finish in time, we'll just continue tomorrow afternoon. And we'll start with an overview of hi-fi reads. And at the same time, we're going to start the assembly, which takes about 10 minutes using hi-fi reads as well. Okay. So I don't know if you have any questions online. Yeah, the chat, there is something in the chat. Oh, yes. So let me go over this. Could you share the video recording via Slack to follow up the practice? Yes, I think we're going to share everything tonight, maybe.
Yeah, it should all get posted onto the BRC YouTube.
Yeah, maybe we'll just send you a Slack notification. And then, Raphael, what were the maximum depth and transition parameters we should have arrived at?
118 and 38, but I can set them like in the Slack.
So it's, yeah, so
114, thank you, and 38.
114 and 38, right?
Yeah.
Okay. But yes, we'll send everything on Slack. Also, all these practical exercise, we have a dedicated tutorial that is recapitulating every step by step. So we can maybe share this as well, so that you have a sort of good.
Some of the stuff's modified though.
Yeah, there is a bit of differences, but just to have a sort of idea.
You want to review some of the theory?
Yeah.
It's interwoven on that tutorial.
Yeah, so we'll share a few materials and see you tomorrow at 1 p.m.